totally agree. I'm gonna crack.
Hi, I'm Christine, by the way.
Hi, Michaela.
Hi,
Oh,
nice to meet you in
nice to
person.
meet you.
Yeah, hope you're enjoying it.
Sure, it's lovely.
So we're 9 to 11.
Hopefully it's a lively
Yeah.
discussion.
It's supposed to be a lively
Guys,
discussion.
first 3 rows.
Right. We're gonna try to get people.
Thank you.
Thank you, Kristen. Yesterday.
right, we'll go start.
As soon as she comes out, you can get started. Just make sure your pack is turned
Yeah, I'll turn it on. Do you want to put the slides up? All right. All right. Thanks to everyone who's made their way back to the day 2 of our Human Cell Atlas meetings. I hope everyone's having a good time in Cambridge. Unseasonably warm weather, but a little rainy. We've sort of been charged with the Human Cell Atlas conversation on heart and vascular. And I will say, and I'll say this probably over and over again, that we have the hardest task because we're given like 2 completely different organ systems. Other than the fact that they both come in direct contact with blood, there's really no difference, no similarities between vascular and heart, different cell types, different diseases. So I think that will be sort of our challenge today. And I want to sort of go through some of the current work that's going on in heart and vascular disease, single cell genomics. And then I want us all to sort of engage actively. I went to the immune session yesterday. I thought it was great. There was a lot of problem solving, a lot of talking about the challenges that exist with single cell analysis for the immune diseases, immune cells, standardization, best protocols. And for us, I really don't think that we're even that close to a first draft atlas yet. So that should maybe be the sort of goal at the end of this, is if we were to have a first draft human cell atlas, what would it look like? So first of all, I guess two announcements. This is being webcast, as you guys all know, on YouTube. For that reason, they were requesting that most people sit in the first 3 rows. It does look bad and not as interactive if people are all the way in the back. And then also, sort of along those lines, is that if you do speak, you will be webcast on this YouTube platform. So those are the only announcements. I wanted to introduce myself. And then before we get started, maybe the 4 of kind of like us co-moderators will also introduce ourselves. But my name's Raj Gupta. I'm a cardiologist at Brigham and Women's Hospital. I did a postdoc here at the Broad Institute with Sate Katharayson on the genomics of coronary artery disease. And one of the projects I worked on, actually sort of the focus of my postdoc, was a SNP that was associated with 5 vascular diseases. And I identified that it had a regulatory effect only in endothelial cells. So in a lot of these sort of GTEx, transcriptome-wide analyses, that association was lost because endothelial cells only make 3% of a blood vessel. And so that got me interested in the single cell architecture of a blood vessel, and that's sort of how I'm here. I've been working with the Regev lab on protocols for vascular tissue, primarily, not as much on the cardiac tissue. And then, we have 3 other disease and methodology experts here. So, Rick, do you want to just say hi to everyone?
Sure, hi. So, hi, I'm Rick Mitchell. I'm a cardiovascular pathologist, also in Boston at Brigham and Women's Hospital. My research interests are primarily in acute and chronic allograft rejection, specifically for heart, but it's applicable to a variety of other solid organ transplants. I've done extensive work collaborating with Peter Libby and others at Brigham, looking at also atherosclerotic disease. So, heart and vessel are no strangers.
All right. Barbara?
Hi, I'm Barbara
Michaela.
Hi, I'm Michaela Nozed. I come from Imperial College in London. I'm a cardiovascular biologist. I'm interested primarily in paracrine mechanisms regulating and maintaining cardiac homeostasis and what happens after ischemic injury. I came into studying to use single-cell approaches because of, to dissect the stromal compartment of the heart. I started with very simple single-cell QRT-PCR, and then fortunately all of these new technologies came along, and I started to collaborate with Sarah, with Norbert in Germany, and we're really trying to bring together and create a human atlas in health and disease. Thank you.
All right, great. Yeah. So I just have a few slides, but again, we'll give everyone a chance to introduce themselves as well. But to begin with, like I said, our challenge is heart and vascular, so two sort of separate organ systems that we're going to analyze together. And we, so fully expect that we're going to have different priorities with each of these. But for the, what is great about heart and vascular is that unlike maybe other diseases, there's this like constant jockeying for who has the best disease. We won't get into that at the session, but for both heart and vascular, there are distinct diseases that seem to be caused by cell types and cell states. And for the heart, that's examples of that are cardiomyopathy, ventricular tachycardia, and congenital heart disease, which is a patterning defect. And so you can imagine that single-cell analysis will have direct relevance to the pathology of these diseases. And then recently there's been a lot of work on sort of the cellular architecture of congenital heart disease, for example. This recent Nature paper from a group in Australia showed that the stages of trabeculation, these kind of ridges in the heart muscle, the actual process is actually ingrained genetically and developmentally in heart cardiac myocytes. So sort of an interesting application of cellular analysis, and that's without single cell techniques. And so using that approach and moving to human tissue will be, I think, very, very valuable. And then for vascular, again, there's a lot of diseases that are just cellular in nature. The canonical example is atherosclerosis, so thought to be the first cellular disease. Rudolf Virchow, right, so the father of the cellular basis of the disease, used atherosclerosis as his first model of a cellular disease. Arterial dissection, hypertension, endothelial dysfunction. And just sort of the example of this is atherosclerosis, we sort of draw these cartoons of a blood vessel wall. And when you get cholesterol buildup in the coronary artery wall, we think there's like 4 major cell types: endothelial cells, vascular muscle cells, foam cells, maybe some immune cells in there. But with single cell analysis, I think this could be further ingrained. We could really understand a process like this. And so the state of the field, where are we? I mean, there's a lot of people using single cell methods. I think like with 10x and other sort of droplet-based methods, this field has been democratized, right? We all can do this pretty quickly now in our labs. But there's been no comprehensive map, I think, at this point. So I'm just going to highlight a few studies that have been done. I mean, recently in Circulation this year, a group in the Netherlands published this paper where they used single-cell sequencing in mouse hearts that were healthy and diseased here meant ischemic, so they caused ischemia, and they used very small numbers of mice. They compared the control to the ischemic, and they identified this sort of new population of cells that expressed CKAP4. That was a fibroblast population, and they sort of implicate that as a marker of this fibroblast population that occurs in response to ischemia. So it could be a very interesting finding. Is it relevant in humans? Is that what's causing disease? They have some nice data where they overexpress this marker, and it does result in increased kind of disease. They can induce expression of the marker. So interesting. And then as far as vascular, I think we all sort of are pointing towards the recent Tabula Muris paper, right? 20 tissues, one of which was aorta. And I will say that, so the number of aortic cells was on the lower end, but, very comprehensive analysis. And one thing that I thought was very cool in this paper is they looked at the transcription factor signatures. So from all their sequencing, they had 1,000 transcription factors in their sort of, 2,000 genes, or more than 2,000 genes in this case. They had a total of 2,000 transcription factors that were expressed. And on the right here, you'll see that, for the endothelial cells, they could sort of assign endothelial cells to their organ of residence based on their transcription factor signatures. So this idea that endothelial cells carry a marker from the organ that they're in. And so in both cases, these were animal models used very well, right? And but you know, this is sort of the state of the field. Is the best maps we have for the heart and for blood vessels has been in mouse models, right? Human atlas will require more work given our access to tissues. And so I think Michaela has some slides she wanted to show as well before we get started with the conversation. But yeah, we'll go from there.
I think we can use this slide just to start the discussion. Doesn't want to be a serious presentation. I think, oops. We've seen it in several other breakout sessions. The first aim is to create a reference atlas of the heart. So definitely we need to do this. The first question is, as in many other organs, is what is a healthy heart? How can we access a healthy heart? So I think it's a discussion that it might be good to start without me imposing or starting with any, telling us what we think. It'd be nice to know what everyone is thinking about this. And then we will build in parallel developmental atlas and developmental atlas and an adult atlas. I mean, the correlation with disease and the importance of doing this is very obvious. The developmental atlas in cardiac disease has an immediate benefit and impact on congenital malformations. And the benefits of building an healthy reference atlas is fundamental to address questions in ischemic heart disease, which is the first cause of death worldwide, in cardiomyopathies, and so on. So I don't know if we want to start to discuss about what is an healthy heart.
Yeah, so first, before we get into the discussion, I kind of wanted to, so, you know, we introduced ourselves. Some of us are interested in heart, vascular. I wanted anyone in the audience, you don't have to, but like if you're working on single cell analysis of heart tissue, vascular tissue, introduce yourself, maybe say what you're working on so we have a sense of the audience.
So my name is Norbert Hubner, and I have been leading over the past 12 months a pilot project within the CZI on single-cell sequencing in the heart. Okay. And I would like to disagree a little bit on your differentiation between vasculature and cardiomyocytes. And, and I think it's very good to do this, but on the other side, there's a lot of interplay. Sure. And I think actually this is super interesting to address also the interplay between the vasculature and the myocardium or cardiac tissue, so to speak, because hypertension certainly also has an effect on the structural morphology of the heart. Sure. Factors like TGF-beta are actually coming from the vasculature and induce a number of changes in the heart. So I think we should capture this as well and not keep it completely as separate entities.
Right, right. Yeah, that's a good point. So, you know, one thing with vasculature is that it probably affects every organ system, including the heart. And so even for the lung, for the gut, for the immune system, the interplay with vasculature will be important. And so for the heart, it's no different. It's a good point.
Hey, thank you. I'm Loyal Goff from Johns Hopkins, and we have already generated a few single-cell data sets in CNS, retinal, and non-CNS vasculature under varying conditions of either ischemic treatment or hypoxia, but also in mouse models where we've done a number of different genetic mutations that are associated with retinopathies or vasculopathies as well. So disease is important, but also the idea that one of the interesting things here, I think, is that we can actually have models that will allow us to look at development of the vasculature in an adult system and in a model system, and those are like hypoxic conditions, for example, where we can induce angiogenesis or vasculogenesis.
Interesting. Are you doing any human work also, or?
Proposed, not there yet.
Okay. All right. Anyone else? No one else? Okay, perfect. So Rick, do you want, oh, go ahead.
So here in the YouTube, it says that Gregor Andelfin from Montreal has been working in mouse and human heart tissue, mostly development, and would like to contribute.
Okay, perfect. So that's from the web server. Okay, cool. All right, so was there someone else? Yeah.
She's not here, but I'm collaborating with Bernice Morrow at Einstein, and she's working on the, she's done single cell studies on the outflow tract and developmental heart in mice.
Okay. So sorry, the CZI project is human tissue, right? Okay.
A number of people involved, like John and Cricket Seidman,
Okay.
and Stuart Cook, and also more on the methodological side, Nikolaus Wajewski, and Jan-Philipp Juncker.
Okay.
And then we teamed up during the course of the CZI pilot projects with Sarah Teichmann and Michaela Nozeda to establish procedures for tissue procurement that are working actually quite well in Cambridge. And we have addressed a number of fundamental challenges. The other Cambridge, yes, across the ocean.
Sure.
And we addressed a number of fundamental challenges because of the size differences of cellular size differences, sure. Large cardiomyocytes versus rather small cells, and multinucleation issues and things like that.
Okay, well, so you're actually, it seems like the only person who's done human cardiac tissue at this point. So, I mean, I think as we discuss more, it'll be really interesting to hear where you are, how many samples, those kinds of things. I think there was one more person who, nope, definitely more people. Okay, go ahead.
Hello. Hi, I'm Xuegong Zhang from Tsinghua University in Beijing. So myself is bioinformatician and supported by a CZI pilot project on the computational side. But I do have collaborations with local hospitals in Beijing working on human hearts. We are developing protocols trying to get the single cell analysis of the explanted heart tissues.
Okay. We would, yeah. No, we'll talk about that. But yeah, right now anyone who's working on something.
We have someone up here too.
Okay, go ahead.
Hello, Mats Nilsson, Stockholm University. I'm part of a collaborative project in Sweden where we're doing a human developmental cell atlas, and we're with particular emphasis on brain, lung, and heart. And we've done so far mostly work on the brain and heart. Okay.
Is that your paper in Cell Sneak Peek on the embryologic?
Right. So that's Joachim Lundberg's part of it, and I've been working with him to make also now spatial atlases of that particular heart tissue.
Okay. So you use developmental hearts at what stages for that study?
Yeah, it's in the first trimester.
First trimester, okay. Interesting.
So Shankar Srinivas from Oxford says that they have been working on embryonic heart tissue in mouse and human, and they are in Oxford.
Okay. Embryonic. Okay, so a lot of developmental biologists who are part of this, and it makes sense. With the heart, the developmental biology actually directly results in disease. Okay, so I think everyone's kind of introduced the efforts that are ongoing. Again, sort of early days, I think, for our tissue compared to lung or certainly the immune system. So, if we were at the end of the session to sort of have a first draft atlas, I think we would have to say, what parts of the heart would we study, and how would we get those tissues? So Rick's a pathologist. He's gonna tell us if that's even possible.
Okay, so I think actually heart and vessel is a wildly heterogeneous, related in many ways, but heterogeneous territory. Certainly in the adult heart, we're talking about atria, ventricle, auricular appendages are even different, valvular conduction system, septum versus lateral wall, RV versus LV, all feeling very, very different effects. Even the vascular system, where you may think that large vessels pretty much everywhere are created equal, we know that there are a number of vasculitides, for example, that affect just the head and neck vessels and don't affect the lower extremities at all, or the viscera. And vice versa, we have a number of other arteritides that affect viscera and don't affect head and neck. So there's something different about the developmental onlog in those different sites. So I think the challenge with the human, as opposed to any mirroring models or other animal models, is also how we procure that tissue. And certainly if I'm holding a heart in the morgue or in the cutting room, something has happened to that patient that has affected the function and the structure of that heart. And we don't routinely, in fact, almost never, biopsy normal human heart. Even if we biopsy the normal human heart, we're looking at a very restricted subset of cells. The way that that is done is a right ventricular biopsy of the interventricular septum. You run in a catheter down the internal jugular and then across the tricuspid valve and into the heart. And it's a random sampling of about 100 microns thickness into the heart. So we're not even anywhere near a lot of the other interesting components. So I think there's going to be a challenge. I think that we should, from the very beginning, as we do this, embrace the fact that if we're using human adult or human well along, not neonatal or not fetal tissue, that there's going to be pathology there. And we should realize and recognize and embrace that and look at that very much in depth. Having looked extensively at the GTEx tissue samples for myocardium, for example, I know that even though they are theoretically from relatively normal, healthy people who've had other traumatic accidents, there are apples and oranges in terms of myocyte hypertrophy, interstitial fibrosis, and working with groups over here at the Broad and the Whitehead, the pathology of the samples that they're looking at a molecular level is really very germane. So I think that we also need to have pathologists, and not because it's going to be job security for me, but pathologists very, very intensely involved in both the harvest and the analysis of what those tissues look like before we just pull out single cells and try to do something else with them. So a bunch of caveats. I'm very excited about the possibilities and what's likely to happen down the road, but I think we have to go in realizing that this is a huge and heterogeneous territory that's always going to have some disease in it. The final point is that if we're going to use cadaveric tissues, there's going to be a lot of variability in terms of when those are harvested. So I have been on the autopsy service on a regular basis. I know that patients typically die in the middle of the night, and we don't even autopsy them until 8 to 24 hours later. 8 hours is remarkably quick. So that's also going to affect the quality, viability of individual cells that we want to look at.
Yes, maybe that would be a point of discussion. So
I can start to say what we have been doing. Sorry.
No, you go ahead.
Yeah. So healthy tissues. First question is brain death, circulatory death. We try to prioritize brain death. Of course, even in a brain death patient is dead, is the heart healthy, but that's the closest we can get. For disease patients, it will depend on what type of disease and which technique you're going to use, but we're using frozen tissue banks for single-cell nuclei and leftover atrial tissue is something that is routinely available, but of course is only atrial tissue. And in our case, our surgeon can take biopsies from the left ventricle in multiple positions. These are very small, and, but that's for sure a very useful resource. For the fetal tissue, we have a centralized system in the UK. It's called HDBR, the Human Developmental Bioresource System, where they collect embryos and fetuses, and then we can get the heart and other organs. For the fetus, of course, as I'm pointing out, there's some phases of development are going to be more easily available. That depends on the ethics and the laws of the country you're working in. And on this point, I like to also stress something that we don't routinely discuss, is like how do we define an healthy fetus and what metadata we should acquire for this. I'd like to know what you guys are thinking. Yeah.
So backtracking to what Michaela was saying about the adult source, I don't know if you remember in the presentation that I gave at the very beginning, there was like a little flow chart on the Cambridge Biorepository for Translational Medicine, which is run by Korosh, who's a transplant surgeon who's actually at the meeting, but I'm not sure if he's in this session. And what he set up is a kind of team or system. It's also supported by Wellcome, the Trans-Zuckerberg Initiative for tissue acquisition from deceased transplant donors. And there obviously, as Michaela said, there are different types. I'm not an expert on this, but the brain death and cardiac death essentially. And according to Korosh, like some of the hearts are still beating, basically where he's taking the biopsies. So the ethics is for 5 centimeter large biopsies. So that
seems pretty fresh tissue.
Yeah,
then.
and they are not suitable for transplantation for whatever reason. So
it
there's
raises
an issue
if they're not suitable
clearly.
for transplantation, they're not normal.
Yeah.
Even brain death with a normal heart has microvascular spasm and catecholaminergic in microinfarcts. So there's, in fact, ischemic myocardium scattered throughout as well. So not perfect normal.
And how have you taken samples for pathology as well to like assess, or no, it just sort of it's frozen tissue for RNA right away?
No, we bank for everything.
For everything.
For everything. We'll do fresh cells, we'll do nuclei, we'll bank the frozen tissue, fixed tissue, so we have an array. I mean, the advantage is if you have a resource like the one that Korosh is running, you got big pieces of tissue, so you're allowed to do this. If we're talking about a small biopsy, you won't be able to do all of these things.
Right. And on the samples that are larger, are you looking at the histology?
Yes, we fix to then look at the histology and always have those metadata. Mm-hmm. When you have tissue banks, depending on what the tissue bank has available, then you will have all the set from the same patient. But the idea for the samples that we have from the Cambridge Biorepository and also from another source, these are donor hearts that do not find a match, we'll collect as much as we can. And trying to record the anatomical regions. And our priority at the moment is to focus on 6 regions.
Okay. We'll probably talk about that next. Yeah,
yes.
Maybe like which regions. If you can't take the whole heart and grind it up. So
no.
Why don't you go ahead?
Hi, I had a question about metadata. And so is it possible to collect information like cholesterol levels or other metabolites from the blood that can help you maybe stratify patients? Is that useful?
That would be really good. I think it would be a really massive task, especially for the healthy patients, because you have to go and collect. It depends what mode of death, etc. We have collected a number, about 20 frozen samples from NDRI, so when I was working here at Baylor. And for those patients, we have some basal metadata from the patients. Stuart Cook, for example, has a big resource of disease samples where he collects a lot of metadata. So I think one would have to really design studies and very carefully plan and working very closely with clinicians to try and collect all of this information that I think would be fantastic.
I would like to sprinkle in a little bit realism to the whole discussion. I think before we start thinking about cofactors and whether we can stratify according to risk factors, I think we have to address a number of very basic issues. I think it would be fantastic if we can agree already on, you know, a set of regions that we collectively want to do, and then things come in like what you mentioned about microspasms upon catecholamine, effects on the heart, and these are overriding effects, I think, compared to risk factors that also contribute. I'm not saying it's not important, but I think we need to get a baseline here first, and of course we should also not be hindered by the fact that nothing is optimal, and we should do the best we can, and nobody is voluntarily donating his healthy or her healthy heart. So that is a limitation we have perhaps compared to other organs. Right. And I think in terms of risk factors, I think we would need to do large numbers to stratify effects here. There are perhaps more important issues like sex and ethnicity that are stronger effects rather than in the first instance risk factors, just as a point for discussion.
I would definitely agree with that as a first pass. Age and gender are going to be your major things that you want to look at. It's always nice to have additional data, and it's beyond cholesterol, it's inflammatory markers, it's a variety of other things. And I think it's also thinking about whether you're talking about cardiac disease or you're talking about vascular disease. And again, we're in a combined group here. But I think your point too about just embracing the fact that these are not going to be normal. In fact, expect that they won't be, and use that in terms of the analysis. If we know what the pathology is, we can actually get a lot more data out of that and make it more useful overall. Yeah. And so I think that, again, having a pathologist involved in sampling to an analysis of what those tissues look like before we even get into the molecular analyses, I think is key.
Yeah, I thought that was a nice point made by the lung group yesterday. So, you know, with the lungs, right, if you just kind of ride behind a bus, right, your lungs will be altered. So if that will get taken up when they do their single cell analysis. So they actually just embraced it. They said that they would have to even start with disease states because that could help inform what they see in the normal state. And I think that'll be the same with the heart, right? So if you know, everyone has some degree of atherosclerosis. If we take people with like actually clinically identified atherosclerosis and identify the cellular architecture of that, then we'll be better able to identify the atherosclerosis that's sort of underlying in people who weren't clinically deemed to have atherosclerosis. And you know, for the heart, maybe the equivalent of that is cardiomyopathy, right? So we all have some fibrosis of our heart after probably age 20 at some point, or you know, in these brain death cases, they probably have more fibrosis from ischemic death. So if we take people who actually have, you know, clear ischemic cardiomyopathy or things like that, and we then use the cells identified in those hearts, we can better understand the so-called normals. I don't know if in CZI, how many hearts, you know, all healthy, some diseased? What are you guys starting with?
So what we have done is extensive work to test it out in animal models, in mice and in rats. And then we have recently switched over to human tissues from the Cambridge Biorepository to test if dissociation protocols and single nuclei sequencing work on these human samples as well as they do in rodents. And we have confirmed this, but we have not started to produce data from in a sense of from different regions because we have piloting what is technically possible, and that we have confirmed. Okay. So interesting. So and then one last thing before we get to the sites that we'd sample. You know, this comes up often at the other sessions. Is like, how many tissues do we need to be confident this was a good map?
I mean, we can all vote, I guess, but you know, is it 300? Is it 100? Is it 50?
Yeah, I don't think it's a good idea if you start voting. For this, I would really actually like to address the computational part, which you didn't talk about at all. And I think Barbara is actually working on experimental design along those things, right? So what I was wondering is how you plan to actually, let's say, make use of existing pilot studies, such as Norbert has, or also animal studies, where you actually get some idea about the variance, to then really inform the next steps, and then to not have this sort of ad hoc discussion, should we have 10, 50, 100 or something like that, different hearts, and how many different areas do we have to sample, but to get sort of closer there. What are your takes on that, and how do you sort of plan or suggest to approach that?
Mm-hmm.
I mean, I could ask you too, because you've done a lot of this work, but no, it's a really good question. I think along with everything that we're talking about, starting from essentially disease state hearts, we have to think about batch effects and just sort of looking at the metadata to figure out exactly what kind of heterogeneity we have. There are the inklings of methods to start to be able to correct for batch effects, but you know, if you have complete confounders in the data, which I assume in heart, given the number of samples that we have and the complexity of disease states that you guys are talking about, we're probably going to have a lot of complete confounders. And that's where we need to be a little bit more worried about what conclusions we can draw based on the disease data we have. Yeah.
Is your observation as well that if you go to single cell level, that heterogeneity actually decreases across populations? Because if that is the case, and I think we do see it in some other studies, not the heart, right? But if that's the case, then you might not actually need to have 300 or you know, these super high numbers. Do you have these observations?
I think at the single cell level versus at the batch RNA level, bulk RNA level, exactly. Sorry, it becomes much more heterogeneous, more complicated, because we have to deal with cell types, cell states change pretty dramatically, expression levels, ischemic time affects transcription. There's going to be a lot of heterogeneity going on across samples, and even within a sample, that I think bulk can often sweep under the rug. There's also actually, and one thing that I realize we don't have on the list of things to talk about that I'm really interested in hearing from people in the audience who've worked on it is the technologies. Are there specific types of technologies and protocols that are essential in the type of tissues, the heart tissues we're talking about, and how can we work with technologists, we being the computational people, work with technologists to really get the most out of those data.
Can I just challenge that? Because I was hoping actually that if we took, let's say, your and my heart cells and threw them together, and maybe also some very sort of severe disease case, although I heard we all have some heart problems already. It's a good take home for me. But don't you think that our cells, individual cells, could be very similar, but just the composition sort of, I don't know, maybe vasculature, you know, like we have more of those different cell numbers. That's actually changing. If that's the case, then, you know, the batch effects, you could do alignment, you could transport labels from another heart. Do you understand what I mean? So it's just more population numbers than actual genetic
absolutely.
states.
So there might not be a change in, there might not be a brand new cell type, but you just get more of a ventricular myocyte or something like that. I think that's what the animal models could be informative, right? Like you said. So you know, I'd say with atherosclerosis, that definitely could be the case, that you don't get like a new kind of macrophage, you just get more macrophages that come in. I'd say with cardiomyopathy, we do think it's actually a transition to a new cell state, like there's a fibrotic cell. So again, like for each disease, it'll be different, but it's a good point. So your point is that if you're comparing just cell numbers instead of cell types, you would actually, the batch effects wouldn't be as relevant. Like
that would be my hope, that this would be an answer to not actually having so many people, but just more cells. Because, you know, that's always the trade-off, right, if you have a fixed budget.
Right.
I want to make a comment about using animal models. To make the animal models useful, then they have to somewhat represent the population. That means much more genetic variability in their background. We can't just look at the same cell lines. So we need to start to look at many cell lines. We need to look at young, old. So I think it's something to keep in mind, and how much investment should we put on that, and how many numbers with the mice, for example. Would that be something that we do in parallel, and so that we can have an estimate for the long term?
I find the aging effects in single cell in general super interesting because I think some labs, I think John's and Claude have shown that the variance and these type of things go up. So very basic effects, but I don't know if there's that the variance of gene expression in general goes up over age, right? John has his one paper, right? And there's I think some follow-ups. I guess the heart there's some sort of more large scale effect as well, but. Sure.
Yeah, there's a few more questions in the audience.
How are we going to address early cell-fate decisions in humans? I mean, we can do this in mouse. Are we going to go for organoids and iPSCs, or can we get material that early, or that's because a lot of these early cell-fate decisions will determine morphology, right? So
right. Yeah. So I mean, this is maybe a developmental question.
In my experience, because I've just been exploring these options, at least in the UK, for us getting anything before 6 weeks post-conceptional week, it's very hard. Is it doable? It's not doable in a matter of months, but maybe our colleagues in Sweden that have done a lot on the first trimester, they can tell us how they got access and what age exactly you were able to profile.
Microphone.
Yeah.
Yeah. So we get specimens from Karolinska Hospital. There is a centralized facility for collect a biobank for collecting these kind of samples, and I think it's the normal. Something from week 6 to 12 or so.
Okay.
Oh, wow.
Yep.
Okay.
So the problem is just before the 6 weeks.
Yeah.
6 to 12 weeks.
6 to 12 weeks. Yeah.
Right.
So those are the tissues more easily accessible, definitely.
Okay. So yeah. So.
Go on
to
one thing we haven't yet touched about is like, we just mentioned, but it'd be good to discuss a bit more about the technicalities and difficulties to sample the entire spectrum, cellular spectrum of the heart, because the cardiomyocytes are really big. I mean, there I put an approximate relationship between a lymphocyte and a cardiomyocyte. So a lot of the platforms are developed keeping in mind the hematopoietic system. So the cells there are very different from the cells that we are dealing with. Plus, they can be multinucleated. So how do we get around that problem? Well, one easy shortcut is just to do single nuclei sequencing. But that is a shortcut, and it gives us a series of answers, but not all the answers. So isolate is a platform that has been proposed to work well with cardiomyocytes. I think Norbert has experience with the isolate. I don't know if you would like to comment about the technical difficulties with the cardiomyocytes and your experience.
Yeah, so it's difficult to address this with conventional drop-seq methods, and so we are trying to address this in different ways. For example, with Oni Basu at University of Chicago. Where is she? Is she here? Maybe you can talk about this yourself. So I cover first ICEL8, and then Oni can comment. And then, of course, well-based methods that are a bit independent of size, cell size, are probably the way to go here.
And you're still using individual cells, not nuclei?
Individual cells.
Okay.
Then. Yeah.
Is there a reason you chose not to go like a
we do nuclei, yeah. But I think, need to do this in parallel because the cardiomyocytes, to capture them, again, with conventional drop-seq methods is difficult. So the combination of live cells plus nuclei, I guess, will give an accurate picture here.
Okay.
Actually, I would also
so just maybe Oni can briefly.
Can you hear me? Yes. We have started a collaboration with Norbert a few months back, about 6 months back, where we started getting adult cardiomyocytes fixed in methanol, and we have been able to successfully load them in drops, custom drop-seq devices, and also we were able to lyse them. The other thing that we should note about these adult cardiomyocytes is there is a lot of proteins, extracellular matrices that go along with it. So they are very structured cells, and lysis was a little bit of an hassle, but we were able to do it. And I should also say that we have some adult heart data also on human and chimp as a comparison, but they are all single nuclei data.
A couple points. Not only can the cardiomyocytes be multinucleated, they are, about 10%. We'll have at least 2, and occasionally more. 2, I think it's also very short-sighted to just focus on the cardiomyocytes. Although they constitute the huge bulk of the heart, of the myocardium, in fact, they're only 25% of the total cells. And the interaction between cardiac fibroblasts, cardiac endothelial cells, and cardiac myocytes is actually very germane and should not be excluded or ignored. So just looking at the cardiomyocytes is too short-sighted. Finally, they're inflammatory cells. They're innate immune cells that are probably very important in terms of normal structure and function. And so recent work from the people at Mass General looking at the AV node have found that, in fact, macrophages express connexins and are critical for normal conduction. So we need to look at everything.
Yeah. Go ahead.
So I just wanted to go back to the batch effect question and whether, I guess I'm not quite sure what we're talking about there, but is the vision that there will be multiple locations where heart's tissues are collected and multiple places where the dissociation's done and the single cell protocols are run? I mean, how many different places across the world are we talking about there? Because, I mean, that might feed into the question of how many hearts or whatever. I mean, it seems like at least it's important to say how are those going to be spread out across different centers, et cetera.
Right, yeah. So I think, I mean, it's going to be done widely. I think there's going to be consortia. There's some already. Certainly there's efforts here at the Broad to do human heart sequencing. And you know, on kind of a first guess, I guess there'd probably be 15 centers that probably work on this just right off the bat, maybe even more. And will there be an opportunity to compare data or aggregate data, or will the batch effects be so insurmountable? And batch effects are also going to be sampling, right? So the heart is not a repeated organ, right? So like the liver or the kidney, where there's different kind of parts that repeat over and over. So you can maybe, you know, even if you sample from different areas, it might be the exact same. The heart, you know, every myocyte could conceivably be different. And so my sense, and people can kind of chime in here, is I think the batch effects will be insurmountable to just aggregate all the data together. I think that will be too challenging. I do think this will be separate efforts, but will be complementary, and hopefully will provide some reproducibility for major disease-relevant markers or major, you know, new populations. But that's just my guess.
I mean, that's a little scary, right? That we can't aggregate across the 15 centers. I think, I guess returning a little bit to Fabian's question too, you know, we sort of have this idea that we have some number of cells, and that makes up for the fact that we only have, you know, 200 parts, samples from a particular region of the heart. And I don't think that there's a really clear mapping yet, at least in my head, between the increase in the number of sort of these independent samples, which is the cells, and the power that you get from having so many of them, especially when you can aggregate across different centers that are collecting the data. So I don't exactly know what the power trade-offs are, but I also think it's worthwhile to actually start developing approaches to make those connections.
So Rick, can I ask a question to Rick? One of my one of our always discussion now we have with Sarah and Norbert, we try to focus on 6 regions. So we focus on the free wall of the left ventricles, the apex, the interventricular septum, the free wall of the right ventricle, and the 2 atrias. So it's, you might say it's quite vague, but what can you tell us? How can we standardize at best the way these pieces are sampled, and how can we then compare? So can we come up with some anatomical points and coordinates to tell us, well, we get that piece as close as possible? And is it something that we feel it might be of any help?
Sure. I mean, I think that once it's decided what exactly you want to look at, then you can somewhat standardize that. And I think a mid-transverse section through the myocardium, so kind of halfway down from the base to the apex, and a mid-transverse section, you could look at lateral free wall, septum, and RV in that plane and do that reasonably routinely standardized. I think that you can definitely look at AV node, and I think that you can look at AV nodal tissue. I think looking at all 4 valves is not as, I think looking at 1 valve, and you could routinely look at the aortic valve. And maybe mitral, I take that back, because there's myxomatous degeneration in the mitral, there's calcific degeneration in the aortic valve, and they tend to be different, so it would be interesting to look at both. And in terms of the atrium, pick your poison. Actually, so it's been demonstrated that there are a greater number of stem cells, myocardial stem cells, in the atria, more on the right than the left side. So I think that would be something else that we would want to go after. But I think there are probably 6 sites overall that you could do. I think that you can standardize it to some extent, and that you could sample routine. But that's holding the heart. If you are doing it by biopsy, I think that now it's much more of a crapshoot. So I think that you are going to be very constrained in the number of cells, the type of tissue you can get, and the depth that you can go.
So actually, as someone who will think about doing downstream analyses of these data, one question that I had for people like you is whether we can develop some kind of coordinate framework, map both living and dead deceased hearts to that coordinate framework, and then reliably be able to place each of the samples on that coordinate system.
So as opposed to an animal model where they're going to be relatively homogeneous, every heart is unique in terms of that. I mean, you can try to get coordinate and map it appropriately. I think that you're going to be off to a variable extent. I think that you're going to have to live with that kind of variability. A little bit of dilation, a little bit of hypertrophy, a little bit of fibrosis, relative predominance of a right or left ventricle. I think it's going to be quite difficult. Even looking at trabeculae versus muscle that is not trabeculated, I think is also going to be different.
So what types of questions about transcription are interesting in the context of the spatial structure, the morphology of a heart? Are there interesting questions about gradients of gene expression or cell type or structure related to the framework?
Well, I think developmentally, I would say that that's absolutely key in terms of gradients. I think that that's going to be a very critical sort of question. In terms of the adult heart, again, it's related to the pathologies and interstitial fibrosis, endothelial cell dysfunction, cardiac hypertrophy that is not uniform. Even in a hypertensive heart, the left ventricle is not uniformly affected by that. So it's somewhat unique depending on the geometry of the heart. Again, as the heart contracts, it doesn't actually, it's not just a tube that does this, it's a squeezing, very complex three-dimensional motion that affects individual cell activity. So even that, the kind of where you are in terms of apex to base, in terms of that squeezing motion of the heart, there are probably going to be gradients of difference in terms of expression of certain myocardial sarcomeric structures.
No, I would add to the interesting pathological changes you just alluded to, also in the healthy quote unquote heart. I think this is already quite interesting for cellular circuitries. If we can do spatial resolution within the heart, which cell is secreting what factors, and that affects the program of other cells, fibroblasts versus cardiomyocytes, for example. And I think we can address these questions with the appropriate analyses in the heart, even without comparing to disease.
So another question this brings up in my mind is we have actually a lot of imaging data of the heart, so live beating hearts, MRI type imaging techniques. What role do those play in this analysis? Can we bring them in at all, or are they just sort of something we can't use?
Well, I think actually having functional data on anything that we're going to eventually analyze is key. So looking at contractility and whether there's non-homogeneity in terms of the wall motion is critical. We won't have that information, however, on the vast majority of what we're looking at. So the exceptional case, we should be all over it and look at every single cell in that heart, but it's most of the time we won't have that data.
So you're saying essentially use it for phenotyping, right? So understand sort of at a gross level what's happening with that heart.
Right, no, to see what the ejection fraction is, to see if there's non-homogeneity again in terms of the wall motion, looking at valvular motion. So I think there are a number of things that we would love to have on every single specimen. You're not going to have that in the vast majority of cases. Even conduction system mapping, it's unique for every individual patient, and that will probably also influence local gradients of transcription factors.
I mean, I will say that it could be possible. I mean, we're talking about a limited number of hearts, and let's say we limit it to all sort of brain death cases where it was rejected for organ transplant. So all those patients would at least have a cardiac echocardiogram, probably not a cardiac MRI, and an echocardiogram. So what information? Definitely EF, and definitely sort of questions about is there any sort of wall that was ischemic or akinetic or something like that. Certainly the reason it may have been rejected for transplant, it could be from the echocardiogram that was done. So that information would be there, but I don't think it would provide like a common coordinate system or something that you could map the biopsy back onto, unfortunately.
Sometimes I think there are other reasons.
Right, of course. There's non-cardiac reasons why they're rejected.
Yeah, I mean, exactly. I mean, sometimes they're complete. So again, I'm not an expert, but my impression kind of from working with these transplant kind of system in Cambridge is that sometimes there are regions that are completely unrelated to the heart, that the people who are responsible for that just don't want to take it, like an infection in a completely different part of the body or something. Right.
So is it unrealistic to try, given the complexity of the structure of the heart, is it unrealistic to try and build a spatial model in a non-human model system and then try and map that? I mean, can we spend concerted effort trying to get that geometry, that coordinate system put together there, find marker genes for each attribute that we're interested? Is that where we should be spending effort at this point?
I would say yes. I think that you're going to be able to have much more control, better quality material, and be able to look at the very subtle things that we've been talking about in animal models. And then you can try to see if there's correlations if you sample wherever you're going to sample in the human to see whether they're kind of you're comparable.
And is there a preferable model system for the structure, physiology, et cetera, of the heart that should be the focus for the human cell
The animal
atlas?
model that's most comparable is pig, is porcine hearts. Those are the most comparable, but actually that ups the ante. Virtually every single cardiac myocyte in them is multinucleated. But physiologically, they're the most human-like.
I know there was someone who said that they were working with, I guess, monkey or primate hearts. I guess maybe not here anymore. But I wonder if those hearts are more, I guess, consistently shaped and sized.
They are, although there are some interesting problems associated with primate aging, that there's an interstitial, I mean, it's great and potentially germane to human disease, but they get a progressive fibrosis, interstitial fibrosis of the heart as a function of age. Okay. So there's pathology that just happens intrinsically. Would be interesting to understand that. But so primate hearts, I think, have their own issues, and even getting them is also a bit problematic. It may be easier to get a human heart than some ape hearts.
Got it.
As a word of encouragement from a computational point of view, I don't think it's unheard of. I mean, we can morph gels from one to another. We can morph images from one another. We could morph common coordinate systems. So if you had relative distances in a single heart, you could then morph it into another heart and then have the positions of the myocytes versus another in some sort of relative framework. So I wouldn't discourage it. I would say that it's going to be just another layer of challenge, right? So.
Mm-hmm. I agree.
I think we should think and try that in some way, because we're always going to be, we are a continuum of differences. No one is like the other, but we could try and rebuild a virtual heart.
And that's some the case.
Yeah.
I mean, there's another organs that we need
Yeah. So we didn't come out with a number like it happened for the lungs yesterday.
Right,
they said
yeah.
100.
Well, they were pushed. They said, well, we don't know, and then someone else said 100, and they're like, great, 100.
So do we feel that the 20 hearts or the 20 donors that was discussed originally in the HCA is going to be sufficient or not? Do we need to aim much further than that?
And how do we actually sample those hearts too? What regions,
Exactly.
how densely? Yeah.
Yeah.
Yeah, so I guess like the sense I'm getting from the conversation so far is that I think we're very interested in sort of a lot of breadth to one sample. So multiple samples, multiple locations from one heart, as opposed to maybe one sample from hundreds of hearts. And then it sounds like we might have to be iterative. So we don't even know what the variability will be from sample to sample. And so that maybe is the more sort of, the less arbitrary answer than like just coming up with a number, is that, you know, as we start to do this analysis, we'll get a sense of what the inter-individual variability is based on age, it sounds like gender, ethnicity, and biopsy procedures, organ obtainment. And we're guessing there's going to be a lot of heterogeneity, but we probably with 20 will have a sense of that heterogeneity.
So maybe instead of keeping going for numbers, we could go for time. Like, let's see how much we can do in the next 2 years, and then from there on, we can, we'll keep reassessing along the way, but maybe I think that's a better point to reassess and really evaluate what people have been doing across the world, because everyone is going to be doing it. And it's important that everyone does it to sample environmental, genetic, and all the sorts of differences that we'll encounter.
Right. At some point, the Human Cell Atlas, there should be sort of a procedure for aggregating the data, and we'll see. You know, maybe it is easier than we think.
So I'm a bit confused because you mentioned about 15 different locations. I mean, I think geographic locations or labs doing these experiments, and now you're mentioning 20 hearts. So I'm assuming now you're talking about doing all 20 in one place. But is that right? Or are you going to do 20 hearts spread out across 15 different places?
Yeah, it's a good point. So I guess we're sort of being loose with this because we don't actually know what's going to happen. But I think that's the truth. But I think for any individual kind of effort to claim that they can create a comprehensive atlas, we would guess it would take maybe 20 hearts to start.
I mean, the 20 organs, the 20 replicates, let's call it, it was what originally the HCA has said. We need about 20 organs to get a rough and good idea of what's going on. So,
okay, so let's suppose we start there and say we're going to have 20 done. There's a question of who's going to do them. Or is that already settled? I mean, are they going to be done in the same place?
I think this comes back to what Raj was saying. people are doing it, so inevitably we'll go more than 20. That's why I'm saying that instead of going by numbers, which should give us time, 6 months, a year, and 2 years, and see how many we got and what informations we got for this, because it's also new that it's very hard to plan and predict.
So isn't the benefit of having meetings like this and discussions like this to try to uniformize protocols across those different locations as much as possible? So, I mean...
So if you take Raj's comments that we would like to have perhaps donor hearts that were refused for whatever reason, to get the largest number in a uniform population would be in major centers that are doing heart transplant. Yeah. So it's got to be, and I can count on 2 hands pretty much where that would be in the world. And then it's just getting those centers, when they have rejected hearts, to acquire as much information and sample, pick 6 sites, and do that in a routinized way.
Yes.
But it's not going to be done in the middle of Kansas and in the Florida Panhandle. It's going to be done in major transplant centers.
Yes.
I mean, I completely agree about standardization of protocols and coordination. So I think what Michaela also said with 20 is what we think within the people we have initiated working with, that this is a realistic number, 20, that we can actually procure over a period of 2 to 3 years and analyze at this stage with the availability of healthy, quote unquote, I still call it healthy, but I completely acknowledge all of what you said. With what we can procure from the sources that we have established access to.
Yeah, hi, over here. This is very far out of my area, but I'm impressed that with hearts obtained in this way and consented for open, that even a relatively modest number like 20 is this enormous resource. And just as you were thinking about doing power iteratively, which is clearly correct, the idea that this resource and keeping them so that you can return again and again to either more areas or an adjacent area, because the platforms are going to change. And if you find that there's more change from, say, 3 centers that are actually making measurements than you can cope with, you can re-strategize and come back to nearby areas and hit them again with just one protocol. And as the spatial things get better, I am, perhaps it's hubris, but I'm confident that you'll be able to do things that you won't do in the first round. So is there a provision in the plan for having these hearts, even after the first sample is taken, to be a resource to return to for the entire HCA heart group?
So I would throw it back out to the audience. What can you do with fixed tissue? So if we can formally fix anything and keep it reasonably well preserved forever, but I don't know what...
But what about cryopreserved freeze after taking the first?
Yeah, I don't actually. So cryopreservation in large tissue like that, even in valves, is extremely problematic. The best cryopreservation techniques do not preserve viability. They do not. So cryopreserved valves, and their cryopreserved valves are thin structures. You would think you'd be able to get that very well controlled. And basically...
So people have actually tried that for RNA, and it's come out badly, because I've seen just the tip of an iceberg of a little bit of data, and I'm not sure it's correct. But so there are things that can be done from fixed, and that would probably still be worthwhile, particularly because all the spatial things work fixed. And so things you learn from wet can be projected onto spatials done later. So that alone would justify it. But the question of whether to try to keep a little bit, either snap frozen works pretty well for many of these things. Nuke-seq out of snap frozen.
Sure.
Yeah, that is definitely doable.
But, I mean, you would only snap freeze certain areas in the heart. Again, you're not going to do the entire heart. So again, it's a sampling issue. I like the idea of being able to revisit the same specimen with improved methodology later on, but we're kind of constrained in terms of doing individual single cell, what you can get out of fixed tissue, I think.
Yeah, but the spatial stuff, you're looking at individual cell level resolution as the techniques improve, and you can probe for,
Sure.
It depends on the number of genes, but it can go very high. It will in the period of this project.
I would like to comment a little bit on the fixed tissue. We have been looking at FFPE tissues a little bit, and it seems because we do typically for high throughput single cell sequencing, 3' end counting, fragmented RNA is not that much of a problem.
Okay.
So maybe I'm being overly optimistic, but it is not such a crazy idea to even think about FFPE tissues in the future.
Other people probably ran across, there was papers on doing this in cancer and formalin fixed tissues, and they could get copy number out of it.
Mm-hmm.
Yeah. So.
Right.
Before we go on, I mean, I get it. The heart's I get it. The heart's the most interesting, of the organs that we've charged with talking about. But is anyone working on vascular tissue at all? Is anyone doing vascular biopsies or samples? And are there certain vascular tissues, or how are you approaching? I mean, certainly the vascular tree is even more complicated in some ways. So how would you approach sampling in that organ?
So you're talking about large vessels.
Any vessel. So yeah.
Yeah.
So right. So how would we go about that?
So I guess we've done some vasculature in mice, and it's primarily been through sorting enrichment of vascular endothelium from reporters, which is obviously not going to be feasible in humans. But I do have a question, if we're diving into this at this point, as to whether the field thinks it's more appropriate, because you have vasculature, obviously, in different tissues that are going to be responsive to different types of dissociation and disaggregation. Do we make an effort to optimize the dissociation for each tissue and then get the vasculature that we can that is probably changed? Or do we make an effort to homogenize our dissociation methods to preserve what we can about the vasculature at the expense of being able to capture different things in different tissues?
Yeah, that's a great point. So I guess for large vessels, and I'm sort of doing this also, so I've done sort of mouse aortas, and I take the whole aorta without flow sorting and looked at all heterogeneity of cell types, and I sort of optimized the dissociation protocol for a large vessel, and I used like a collagenase hyaluronidase dissociation for 30 minutes, and then the reviewer said you have to use elastase, so then we tried that. I mean, so even when you so-called optimized protocol may not be optimized for every cell type, even in a large tissue. So I guess my answer is that I don't think there's much value in standardizing these dissociation protocols, because different protocols will have different value. And so I think you can learn some things from some organ optimized dissociation protocols. So I think that's the fun part about anyone who's interested in vascular, is that there will be vascular tissue from every other sort of single cell effort that's done, including the heart. There'll be endothelial cells from the lung, the kidney, from every tissue. And so analysis of those endothelial cells will give us some look at the small vessels, maybe even some medium-sized vessels, and then large vessels can be done sort of separately with different protocols.
There are, as you mentioned in the introduction, fundamentally different properties in different types of vasculature.
Right.
And if this is passenger information from some of the other atlases that are being generated, I mean, it's perfectly confounded. It always will be perfectly confounded, and there will probably be things that will not be comparable in those contexts. Should we have a sub-aim or a sub-goal to try and have a standardized collection method or focus on nuclei again, enrichment is not possible here.
Right.
I don't know how that problem is solved.
Right. Yeah, it's a good point. So when you say standardized protocol to get the most endothelial cells, for example, from a
not necessarily the most endothelial cells, but at least a consistently stressed population of endothelial cells from across the human cell atlas.
Sure.
Because otherwise, we will always be perfectly confounded by the protocols.
Right. If we're comparing the liver and the kidney, but they use totally different methods, then we can't ever compare those. That's a good point.
I think, sorry.
Go ahead.
I think exactly what you said, that the first step is getting the nuclei. So the protocols for nuclei isolation is quite uniform across different tissues. Then you could enrich for the endothelial cells. As with a protocol or antibodies or whatever you can that can be maintained as uniform as possible. And then you go back with spatial transcriptomics to verify what you have found. So you need, I think, a multiple approach to evaluate, identify the location, and validate what you have found.
So also just to extend on what you said about the spatial point here, should the vascular atlas be the encompassing coordinate system? I mean, is that the place where we should build the full coordinate system for the entire human body atlas?
I personally think there should be at least 2-3 techniques that are uniformly used. I'm talking about nuclei versus spatial transcriptomics that are the same across, because technically they are the same. The processing of the tissue is the same, so that would be a really good way to uniform and be able to compare more easily across organs.
Yeah, I agree. I think that's exciting, is that the common coordinate framework could be endothelial cells. I don't know if any other cell in the vasculature, but endothelial cells are known to sort of have a resident signature. I don't know if it's going to be that exact. Like, for example, if I gave you a biopsy of tissue and you analyze the endothelial cells in that biopsy, would you be able to tell exactly where it came from, or would you be able to say it's from the heart, and that's what you can tell? I don't know. I mean, that'll be something that we could test. Like, could you tell me that those endothelial cells were from the LV? Maybe LV endothelial cells are different than RV endothelial cells. The apex of the heart versus the septum of the heart, probably, I don't know if you'd have that granularity just from endothelial cells.
Well, the study in the brain have shown very
sure,
nicely. There were
true.
gradients of endothelial cells, changes in the smooth muscle cells in the pericytes. So the study in the brain really gave us an idea that there might be even more heterogeneity than we were thinking before, because a certain level of heterogeneity at the microvascular level is there, functionally, morphologically. I mean,
correct.
You guys know better than...
Well, and also, as you're aware, even in the same general location, flow differences in shear stress forces, turbulence, nonlaminar flow, also influences gene expression dramatically.
Right.
So you could be a millimeter away and have a very different signature, even in the same general territory.
Right. That's a good point, too.
Not necessarily a different type, but a different state, to just reinforce what you said.
But I think any sort of vascular project should sort of investigate that. Can endothelial cells or vascular cells serve as a common coordinate framework? I think that would be an aim.
So the discussion made me wonder whether there's any, I think this is really challenging from a practical perspective, but any attempt to try and sample the same organs different organs from the same people, talking about the relationships between different organs. I mean, I'm assuming that you get a heart, and that's what you get, and it was rejected for transplant or something, and so you do experiments on that heart, and you don't have access to everything else. But I mean, you could imagine great value in a data set where we had organs from the same people. So again, it's really a practical question. I've got no idea what's even possible or what the easiest way in there would be, but I was wondering if it's been thought about.
Maybe Sarah, can you tell us in the case of Korush, how often you manage to sample organs from the same patient?
Yeah, I mean, that's routinely the case from the same, I mean, again, that ethics at the moment is for everything that's accessible through the transplant incision, which is from the neck to the waist. And it's basically anything that's not transplanted, so that gives you an idea. Yeah, and we routinely have multiple tissues from multiple organs and so on. Does that answer the question?
Would you say half of the patients you will get 5 or 6 organs from the same patient, or 90%? I'm just curious of the numbers.
No, I mean from everyone you get.
will manage.
Yeah.
And I know, Orit, there's some efforts here at the Broad to sample multiple organs from single donors, right? There's
Thank you. So I work very closely with Kristen Ardley, and we're leveraging actually the previous GTEx pipeline, which is based on NDRI, and it's a very nice pipeline for deceased donors, both for organ transplant and for rapid autopsy. And you can get several different organs from the same donor, for sure, and they have quite a lot of information and metadata that's tracked very nicely. So it's a very nice and robust pipeline that would be very useful, I think, to use. And Barbara, if you want to add more to that, that would be.
Yeah, I completely agree. I guess in my mind, one question that's coming up while we're discussing multiple organs in the same individual and also vasculature more generally is whether we're going to take vasculature samples across, let's say, one individual, a GTEx individual, for example, for the express purpose of mapping vasculature in an individual, or whether we're just going to get vasculature as an offshoot of collecting another organ. So is this going to be sort of residual data, or is it going to be collected in its own right? And from my perspective, in terms of analyzing the data, it would be, I think, very challenging if it were part of a specific organ collection, if it were sort of the residual data that we got from organ collection, because like everyone has been saying, the protocols are wildly different. The individuals are probably wildly different unless we went to GTEx, et cetera. So do you have thoughts on that?
Yeah, so it's not something I thought about until today, to tell you the truth. The human tissue that we have access to, I haven't been getting it from autopsy samples. So I'll tell you, sort of, I'm interested in the aorta. So cellular heterogeneity in the aorta is interesting, and one thing that we had thought about is when people get coronary artery bypass surgery, they have punch biopsies taken in the proximal aorta, and so those punch biopsies are discarded tissue, and so that's a source of tissue that we could use for single cell analysis. At the Brigham and Women's Hospital, there's 300 coronary artery bypass surgeries done a year, and they do 3 of these punch biopsies on average. So that was one source of tissue, but there'd be wide heterogeneity. I mean, different locations. They're sort of chosen ad hoc by the surgeon. They're not sort of sampled, based on our recommendations. Also, there's valve surgery that I've sort of talked to Rick about. Every time a valve is replaced, there's some proximal aorta that's excised with that. And that could be a source, but that's again diseased tissue. So that was sort of my, sort of sampling of vascular tissue, and so it was sort of dedicated to getting vascular tissue, but it was at like one point in the vasculature, not sort of the whole vascular tree. It is exciting. I think you could ask different questions by sampling the whole vascular tree. And so you could use residual data, and there's technical artifacts, but I agree that maybe the nuke-seq would be a way to get around the technical artifact, because that is a sort of consistent protocol to get nuclear, and vascular cells don't tend to be multinucleated or have kind of caveats from using nuclear-seq. So that could get around that. I think that would be more feasible. There are only going to be a few instances where we could actually sample kind of like from the aorta all the way down to its branches, all the way out to the capillaries from a single individual. I mean, that'd be extensive sort of surgical dissection and processing that I don't even know if sort of something's been done like that.
Not on a routine basis.
Yeah, or I mean, even bulk-seq RNA methods, right? I mean, I don't know if anyone's done an experiment like that using bulk-seq.
Not that I'm aware of.
Right, yeah. Just sort of maybe technically too difficult. All right. We've sort of glossed over some of the computational hurdles, but you're interested in batch effects in particular and methods to sort of validate that. There's a lot of computational people here. Do you want to sort of take a few minutes to kind of talk about your approaches or questions that are outstanding that interest you? Barbara?
Yeah, sure, absolutely. So, I mean, Matthew's also in the audience, and he is also he is an incredible statistician in the sense of thinking about batch effects in these particular data sets. So I hope you'll speak up at some point. But okay, so I guess broadly, if we're talking about vasculature or heart or either way, I think one thing that comes up in particular with respect to the heart, for me at least, is the idea of an atlas is not just a description of points in in the tissue space, but really getting at spatial resolution. And again, when you think about the heart as an organ, it's a very dynamic organ. It obviously moves in these very interesting and person-specific ways. And there are going to be interesting stories to tell with respect to disease about dysfunction in this organ with respect to the dynamics, right? So I guess more than other organs, I'm really interested in thinking about this atlas as a spatial, as a dynamic spatial description of the organ, thinking about transcriptomics, but thinking about all the other aspects that come along with it. And in that regard, I'm struggling thinking about 20 post-mortem hearts, thinking about how we're going to do this properly. I realize it's a really hard task, and the statistical methods don't entirely get to the point. But I'm hoping that 20 is going to be the beginning, because I feel like we need to prospectively iterate on the design of the study, given heterogeneity, given the goals of the project, where I think, again, with heart in particular, we can flesh out really what we mean by atlas spatially in an exciting way. Given the specific types of samples that we get to, whether it's mostly male, mostly older, whatever the phenotypes that we get, make sure the stratification is there. So in terms of batch effects, I think along with this perspective idea, we need to take those into account very carefully, be clear about what information we're not going to get from those 20 hearts, right, because of batch effects, and then do a very careful job of adaptive design of the choices of hearts that we make moving forward to minimize that, to minimize the batch effects, to maximize statistical power in whatever objective we have for the atlas, broadly writ. Do people have thoughts about this? I don't want to, I feel like I talk too much. Yeah.
Yeah, so I mean, Barbara, and I'm not sure if Matthew said anything, but I completely agree on the need for thinking through experimental design, standardization. I think that the discussion about kind of beacons and markers spatially is super useful, actually, and that will be extremely helpful in this context. As I said yesterday in the gut, I'm actually feeling kind of more optimistic about this. I mean, anyway, I come at it from the experimental design from this angle anyway. I am feeling more optimistic about batch effect or integration methods, let's say, because I think the computational community is making huge progress on that front. On the front of in terms of what you're saying about skew of donors, just from a practical point of view, I mean, it's going to be hard not to avoid that. Speaking like from the program of that we work with, it's definitely skewed towards older. I mean, there are young transplant donors, but because people die, people have accidents and die at all ages, but it is definitely skewed towards older. And then the other thing, it's definitely skewed towards male for some reason, and I think that's to do with consenting by the families. So the families just tend to, for some reason, like it's biased towards men or the families consent, basically, as opposed to women, and it's definitely statistically significant and quite surprising.
That's true.
Yeah, so GTEx is 2/3 male, 1/3 female, and my understanding is that's because men consent their wives less than husbands. So men consent their wives less, even though the exact same number of donors were
Husbands are disposable.
requested from both genders. So, yeah. Do you want to say something about that?
No.
Okay,
No,
so
but it's correct.
it's and actually also looking across GTEx too, just because that's what we have experience with. I think also the mode of death is really fascinating. The gender skew is interesting. The age distribution is fairly bimodal, actually, right? It's sort of. I'm trying to remember. It's skewed towards older, but we have some cutoff too. The oldest is
70. I think.
70. Yeah. Okay. Yeah. So I think this is interesting in the context of thinking about how to incorporate stratify, especially with only 20 hearts. Something that actually comes up too in GTEx that we've seen is that maybe this is too far in the weeds, but if you have multiple sites from a single individual, so we have on average 18 different samples from a donor in GTEx, it actually turns out that when you think about sort of cross-tissue mapping, I'm thinking about this specifically with respect to vasculature now, you have donor-specific confounders that you really need to control for as well. So it's not batch, but if this person is a smoker or whatever their lifestyle has been, you're going to see signatures of that across the multiple samples from the same individual. And taking that into account is actually a really important thing to do without removing the signal from this the heterogeneity from this donor more broadly. You have to take that into account, though. I want to go back to Sarah's point about experimental design and sort of how we can start a conversation between the experimentalists and the statisticians. How is it best to talk about designing these experiments prospectively, choosing donors, matching samples, and deciding on protocols? How can we start doing that? Bad question. Yeah. So do people have thoughts on that from the collection point of view or the protocol point of view? Are there specific things we need to try out with respect to any of the protocols? Are there specific patient types that we need to get at immediately to try and understand disease and the atlas more generally?
Well, again, I think that if you're looking for the best quality material, that you're looking at a rejected donor. So I think you're looking at a particular, and you take whatever you get. Because you also want all this other additional data, you want to have the metadata, and you want to be able to look at functional data, and you want to be able to put the whole thing together. So, it would be nice to have 20-year-olds who all die on the same time, and we can look at their hearts in a standard way. I don't think that's ever going to happen.
Yeah, I'd say that, so some things that I think this session we could think of standardizing are the type of heart. So it sounds like there's a broad consensus of sort of, post-mortem, not sort of clinical biopsy samples, but post-mortem hearts, brain death as the sort of, or rejected organ transplant, brain dead and then rejected for organ transplant.
Right.
Acknowledging that there'll be some disease, but that's as good as it gets. Then we could standardize sampling sites. And then as far as tissue preparation, so right now I think the sort of standard seems to be nuke-seq for clinical samples, given that you can flash freeze, you preserve RNA, you get around the issue of different cell sizes and sort of issues with that. There probably is a role for also doing cellular analysis, it sounds like, for maybe you get different data from that, but it seems like maybe at least those things, the type of heart, the sampling sites, and the tissue preparation seem relatively standardized. I also wonder if, there will be groups that are focused on the normal heart. There are going to be groups, a paper is certainly going to be published where they compare dilated cardiomyopathy to normal, or hypertrophic cardiomyopathy to normal. And those will probably not be exactly as standardized, right? Because those will not be healthy donors, and those will be the explanted hearts that are going, the patients who are getting transplanted in the explanted heart. There'll be much more variability in those samples, and I think we'd probably just embrace that as it'd be an interesting study. It'd give perspective on the normal hearts, but there would be inter-individual variability that would exceed what we would imagine from normal hearts.
So ideally, those would be the two extremes.
Right.
No, ideally, I mean, theoretically, they would be the two extremes.
Right.
And then we need to find a way to build what's in the middle, because it's good to know what's at the end stage when you have the transplantation, but you think you want to know what led to that if you want to stop it. Mm-hmm. So then going back to biopsies, and as much as we can, it will be.
And at the same time, I think that some of the comments that were made earlier, that we should really take advantage of animal models And because we can do those in excruciating detail and get a lot of information and look for correlates in the human hearts when we're looking at, and then think about what we can do with fixed tissue that we can go revisit with additional techniques later on.
So a related question that I have is, what's the role of differentiated iPSCs here? Do we care about developing those systems and using them in the atlas?
I think it's a really interesting question. I mean, there are groups that are doing that. The Seidman group has been using iPS cells and differentiating the cardiomyocytes. It's only a part of the story. And they're not perfect correlates, even when you engineer in the same mutations. They do not behave exactly like it does in the context of a heart. So I think that you would lose information about fibroblast interactions and endothelial cell interactions, the total metabolic environment. I think there's utility in doing that. I think it's really interesting to do it. It's more manipulatable, but to make that the gold, it's not even a bronze standard.
I think it's manipulatable, sorry, but it's also, on the other side, it would be good as a drug screening and
potentially.
other type, potentially.
Yes.
Potentially.
There are
I mean,
caveats.
for us, it's the best thing we can do in vitro at the moment. It's the,
correct.
In terms of human, it's human iPS. We know they're not well differentiated, and there are a lot of problems starting with.
Right.
But it's the closest
exactly.
that we can, and then we can move on and build organoids and bring in the other types of cells. So there is some space.
If I may add to that, my name is Gert Vingstra from Nijmegen. We're working with Robert Bassier also to differentiate those cells towards cardiomyocyte lineages, which in the process also generates other cardiac cell types. And they follow a sort of normal developmental path, going through mesoderm and then to cardiac progenitors. And the goal is indeed to get to disease modeling in a variety of ways. And you can manipulate the cells, obviously. And you also can generate engineered heart tissues out of that. And so one of the goals there is also to get to more mature stages of in vitro differentiation. And for that, the comparison with in vivo tissue is really important. So I think, yes, there will be some insights that come from that as well, but it is a two-way street, in effect.
So is there someone doing both at the same time, in vivo tissue and differentiation, so that they're done kind of in the same lab to make that comparison?
Yeah, I was just saying that we're aiming to do that, yeah, to do it at the same time.
And how do you compare, how do you think about cross-analysis of the two?
In terms of cross-analysis, we try to generate the single-cell RNA sequencing data set. For the heart tissue, we're not there yet. We probably have to move to nuclei for that. But then we also would need to generate the nuclear data sets also for the in vitro tissues.
I would like to add that we had a CZI project for the Human Cell Atlas, the pilot round, where we did iPSC-derived cardiomyocytes, single cells, single nuclei, and then adult human heart tissue, single nuclei, just to do this kind of a comparison. And the data should be going up pretty soon. The one thing that I wanted to add was, if we are going to do heart tissue from many different sources, can we please note RIN scores if possible? It's not easy, it's not fun, but it is very, very useful in measuring that.
All right, so yeah, I mean, I think we have, what, a few more minutes left? Yeah, 15 minutes, 10 minutes. So maybe with the last 10 minutes, we'll say that, sort of summarize some of the things we've discussed. And then I did want to say that, though we've sort of focused on the challenges, I think, heart and vascular are sort of unique organs in this sort of single cell program. And I touched on this at the beginning, but there are some specific advantages to studying these organs that I think other organ systems, don't have the luxury of. I'd say that, first of all, with vascular, I think there is this prospect of it being a common coordinate framework for the whole body. we will get data that's informative from every sort of effort that comes of this. And I think that's exciting. I think that's something that we could take advantage of. Also, so with vascular, it's one of the few organs that's so plastic. So, the immune system's like this, but the vascular cells respond to local stimuli like shear stress or flow or inflammation or diet even. And so that, sometimes we wonder about how do we integrate exposures when we look at the transcriptome. And a challenge and an opportunity with the vasculature is that we could do it with vascular cells. So if we got information about things that people were exposed to, whether or not it's infections or diet or risk factors for disease, that could be, we could measure that from single cell analysis of vascular cells. So I think that's exciting. And then again, just sort of touch the diseases that we're studying with heart and vascular are diseases of cell state, diseases of cell patterning. And so, congenital heart disease, I think, will be an immediate sort of application of some of the developmental work that's being done in this room. And then again, so while heart tissue is sort of tough to obtain from clinical biopsies, vascular tissue is like ubiquitous, right? There's lots of vascular tissues that are biopsied or excised or come with other tissues when they're excised. So another sort of opportunity, I think, for single cell efforts, if they can be standardized, of course. So those, I think, are unique opportunities. I don't know if there's other sort of unique things that have drawn people in this room to the heart in particular or the blood vessels in particular. Was there any sort of reason that any of you latched onto these diseases or these organs? Maybe not. Or just assigned to this room or something. Yeah, this is the biggest room. So yes, okay. So go ahead.
So related question. I hope someone should say something. But are there specific technicalities or challenges that the heart and vasculature pose that we haven't covered in the last 2 hours? Anyone have anything in particular in mind from their experience?
Yeah, right. Like in some ways, our challenges are the same as every organ, right? So standardization of protocols, batch effects, inter-individual variability. I think our unique challenges with the heart we've discussed are that it's not a patterned organ, so every cell could conceivably be different than every other cell. So even our sampling methods will have to be even maybe more exact than they are for other organs. Other sort of unique challenges that we've discussed in this session are the fact that every heart's going to have disease. We'll never get our hands on a truly healthy heart, unlike maybe other sort of other organs where it sort of comes with a tumor or something like that. And then I think those are kind of the main unique challenges to the heart. Go ahead.
Yeah, so my question is about the type of research people will be doing downstream with this. And again, since these will be a lot of diseased hearts, is there a type of metadata that might be more important for, say, the heart than any other type of organ based on the type of research people might be expected to do with it?
Right, yeah, that's a good point. So if we initially just sort of get a collection of so-called healthy hearts, the metadata that we could then kind of go back and look at would be pathology, I think, would be the best, right? So if there was concomitant pathologic investigation of that tissue with single cell, then if you find some sort of interesting signal, right? There's this cell type that's not in the other samples, then I think the first step would be to go back to the pathology and see if there was something that could be, examples of this could be fibrosis or, some hypertrophy, things like that could be found. There's preclinical imaging, so like we said, echocardiogram, but cardiac MRI, you could, again, find hypertrophy or fibrosis. It's just sort of a different kind of pathology there. And then beyond that, those are I guess.
Yeah, is whole genome sequencing something that we have to keep in mind and not to forget? So if we don't have the resources to sequence right away, we should keep some aside and make sure that if we need, we can go back and check for that as well.
You were asking about potential unique features of the heart. I
I'm not sure if it's unique. Well, it is in my heart unique anyway, but plasticity, I think, depending on load conditions, hypertension, myocardial infarction, pharmacotherapy, ACE inhibitors and whatever, all of these things will affect the transcriptome.
Correct. I mean, in terms of metadata, I mean, the regular cardiac risk factors, hypertension, length of hypertension, therapy for hypertension, cholesterol levels, HDL, LDL, inflammatory status, whether that's a CRP or something else where we understand that. So I think that there's a lot of clinical data that would need to be correlated because, again, there is plasticity that's dependent on what we do, what we take, what we eat.
I mean, sometimes a failing heart will recover if it's unloaded for a period of time.
Correct.
So.
Correct.
I would like to turn the question also around. I think the heart can also learn a lot from the atlasing efforts that are going on in the other tissues. And for instance, I mean, we have talked about fibroblasts. It would be crucial to actually compare what makes the cardiac fibroblast different from the fibroblast in the kidney or in the skin. And I mean, you could even perhaps name all of the organ systems that have, to some degree, fibroblasts within them. So I think we also can inform specific organs by putting the data together and look across the different organs, and the same you could do with macrophages and many other cell types that are not specific to the heart.
Correct. Actually, that's a really interesting point. And in the same way that endothelial cells may provide something that allows us to look at different places in the body, fibroblasts may allow us to also ask some interesting questions because they're going to be in every tissue.
Mm-hmm. Mm-hmm.
And innate immune cells are going to be in every tissue, but they have maybe different functions.
Mm-hmm. Yeah. All right.
Good.
Okay. Well, I guess we'll go on our break. Thanks to everyone for coming. We have a wrap-up session where we'll try to make some sense of this session and sort of summarize the points. But I'm happy to have met all of you, and enjoy the rest of your conference.
Hello, everyone. Take your seat. We're beginning.
So hello, welcome back. You hear me well? Okay. So it's my pleasure to be here finally. And it's my pleasure to chair the summary breakout session. We have 3 interesting topics dealing with the heart, kidney, and liver. And we start with the heart.
Right, thanks. Okay, great. The timer's on. Well, wonderful. Thanks again for everyone who's made it back for the second day. My name is Raj Gupta. For those of you I haven't met, I'm a cardiologist at Brigham and Women's Hospital and a researcher until recently at the Broad Institute, but now at the Brigham. And I've sort of come to single-cell analysis differently than maybe a lot of people. Sort of interested in genomics and sort of was using GTEx data to identify eQTLs that were relevant to disease. And in that, we saw that cellular heterogeneity was going to be important to explain. And so, that's my background. And we had a great session for the heart and vascular session here in the auditorium. We had a lot of people who came. And I'll say that right off the bat, there was a little controversy over the statement, but I do feel that the heart and vascular are 2 entirely separate organs, right? So it was a bit challenging for us to sort of talk about both, given that they both hold blood or come in contact with blood, but beyond that they have different cell types, different diseases, and so we sort of kind of wandered through talking about both of them. But I'll say that at the beginning, to summarize our session, we had the benefit of a really great audience of people from around the world, all of whom are interested in single-cell analysis of the heart or vasculature in some way. I'd say it was about 90% heart, admittedly. I'm more of a vascular person, so feeling a bit lonely. But complementary efforts from a CZI consortium, the University of Chicago, Europe, Asia, Australia, and then even here locally there were some groups. So great representation, and there was a lot of interaction on some of our challenges. And so before we sort of went into the complaining about how this was not going to work, we sort of summarized the state of the field for the heart. And we say there was 2 major themes, I think, at this point, though this is rapidly changing by the minute even. But first, there have been a lot of papers that have found disease-relevant subpopulations in animal models. And the animal models have been incredibly informative because there's less intra-individual variability. The disease processes are more exact, I would say, than we see in the human. And so there have been some interesting studies there. And then there have been some recent insights from developmental biology. And this is in human in many cases. So just to summarize, this is probably a review for many people here, but a few papers that showed disease-relevant subpopulations. This was a paper that was published this year in Circulation at the top, where they took ischemic mice. So they induced ischemia in mice, and they got ischemic regions of the heart and did single-cell sampling. Sorry, here. And what they found is that there was this unique subpopulation of cells that had mostly red dots, which were from the ischemic mice, the 3 ischemic mice. And they found that that population expressed more CKAP4, which they sort of identified as a new modulator of fibroblast activation in ischemia. So, the next step would be to validate this in human studies, right? Is that same population? Are those same genetic modifiers relevant to human ischemic disease? And then sort of a second great paper on the vasculature, also in mice, was from a molecular atlas of cell types and zonation in the brain vasculature. And that was in Nature, where they were actually showing that for different stages along the vascular tree, all the way from big vessels to medium-sized arterioles to capillaries, then back to the veins, there was a gradation of gene expression, suggesting that the vasculature could sort of serve as a signpost for different parts of the body. So that's the sort of disease-relevant subpopulations from animals. And then there were some recent insights into developmental biology that we had a representation of these groups in the audience. It's from Stanford. They recently did a single-cell analysis of progenitor cells in coronary artery populations. And then right off the, and not even hot off the press, not even made its way to press, is a paper from a group in Sweden where they did expression analysis in developing human hearts. And by developing, they say sort of embryologic age 6 to 12 weeks, which I find fascinating, and we'll sort of delve into that paper more. So, okay, so the challenges that we discussed in this session were many and a bit overwhelming at times, but I'd say that they were probably similar to what the other groups discussed as well. Maybe a few things are unique to the heart in particular. So first of all, we spent a lot of time talking about standardization of tissue procurement, sampling, and dissociation protocols. And so that doesn't really exist for the heart yet. I think, from our audience, just of the people here, there were maybe, at least 10 groups who are working on this. And we haven't discussed, are we going to use nuclear sequencing or cellular sequencing? Are we going to use certain regions of the heart? So I think that right now there's no standardization, and that's sort of a big task, I think, for our working group going forward. And again, sort of similar to other organs, that'll be the case. But for the heart in particular, there's no sort of repetitive structure, right? So, at the last point here, as I say this, it's a non-repetitive organ. So there's not like zones of the heart. Every cell in the heart could be different from every other cell. The apex could be different from the septum, even within the same anatomical structure. And for that reason, we might have to be more exact about our standardization. We might have to have better coordinates so we can directly compare cells, or we could sort of say, well, forget it. It's going to be too hard. We'll see what comes out of it. And I think our consensus was that we should be more exact. I think that was the consensus from our group, and I'll talk about that in a second. And then, the second sort of thing is this problem of inter-individual variability. So what metadata would we collect? So certainly disease. So, and we'll talk about this. There's actually no such thing as a normal heart that we will obtain. So even in these sort of cases of brainstem death donors who have presumably normal hearts that are then rejected for organ transplant, even those hearts, because of brain death itself induces some microvascular ischemia, some coronary vasospasm. There will be microinfarcts in those tissues. There will be diseased segments. There's always subclinical atherosclerosis in anyone over the age of 20. So we will see disease in these tissues. So we'll have to think of variables to record so that we can then sort of after the fact determine that. And then obviously gender, ethnicity, and sex. And there's other sort of clinical markers we could record as well. Inflammatory markers, lipid levels. But we thought these would maybe be the 4 things, at least on a first pass, to focus on. And then I sort of mentioned this tissue access. So we spent a lot of time on this as well. And so if we were going to have a standardized way of doing this, if there's no such thing as a normal heart, how would we sort of account for this? And we thought, well, you could either go with post-mortem samples or clinical biopsies. Clinical biopsies are pretty rare from the heart. We would never biopsy a normal heart. And even the diseased hearts or post-transplant hearts that we biopsy, we take such a small piece of tissue and are never really sure where that biopsy is from. The way it's done is that you sort of snake a catheter into the internal jugular vein into the right side of the heart and just sort of blindly use these forceps to grab little biopsies of tissue. It doesn't sound so inspiring, I guess, to describe it like that. But I think that would be a very difficult tissue source for this kind of community to standardize. So we focused on these post-mortem hearts. And even though I sort of told you all the problems with using rejected organ donor hearts, that did seem to be the best sort of first pass for our group. And so we just have to account for preclinical disease. And so we can think about ways to do that. And that might be collecting medical record information, and it could be pathology samples. So at the same time that the heart is taken for single cell, it'd be important to also use some sample for formalin-fixed tissue and look at pathology so that if we see sort of variability, can we go back to the slides and see if there was more fibrosis or more hypertrophy and those markers of disease that might not have been clinically apparent but will be pathologically apparent. And so, and then for vascular tissue, again, I'm a little jealous. The heart got 90% of the discussion, and I'm sort of more interested in vascular tissue. But I would say this was easier. Maybe that's why we didn't talk about it. But for vascular tissue procurement will be easier in some ways because we do biopsies from different parts of the vascular tree. Aortic biopsies occur during surgery. When we take out the aortic valve during aortic valve replacement, we get some aortic tissue. And then also there's this sort of opportunity / challenge of there will be sort of residual vascular tissue that comes along with everyone else's organ? And should we have like a protocol for studying that? I mean, everyone will use different protocols for tissue isolation, but I think, as a vascular interest community, I think that could be one of our main challenges over the next year, is to think of a way to take all the endothelial cells that are obtained from other tissues and reanalyze them in some sort of meaningful way. And so then, so specific challenges to the heart. And so this actually I found to be quite interesting is that, fine, let's say we can dissociate the tissue and we can sort of have a standardized specimen and a standardized protocol and a sampling region that we're comfortable with. The problem is that 10% of adult cardiac myocytes have at least 2 nuclei. So even if we're using a sort of multi-nuclear, or we're using nuke-seq as our standard protocol, we would probably reject that 10% of cells. And these cells might be very relevant to regeneration or disease progression. And so that's sort of a challenge that we might have to deal with, is that how do we sort of capture multinucleated cells if we're using nuke-seq as our standard protocol. And then again, cardiac myocytes are only 25% of heart cells. And so some of the challenges that people voiced are that cardiac myocytes, using cellular analysis, is almost impossible. They're too big to sort of use through a standard droplet 10x system. But perhaps we could do nuke-seq with some cardiac myocytes, or but a different protocol for other cardiac myocytes. I think for me, this sort of emphasized that if we're going to have a comprehensive atlas of the heart, it's going to have to include many different dissociation protocols. It won't be sort of one protocol that works for everyone, is sort of my sense from some of these challenges. But it wasn't all sort of doom and gloom. We saw a couple unique opportunities as well. So first of all, we spent a little bit of time talking about if vascular cells could serve as a common coordinate framework for the whole body, right? There's recent papers from the Tabula Muris group where they showed that like the transcription factor signature for endothelial cells was representative of the organ that they came from. So that sort of suggests that there is a signature. The problem is endothelial cells are also pretty plastic, so even 2 cells next to each other, if they're exposed to different flow conditions or different lipids, they might be different. So we don't know if this will take some method development and standardization, but if any cell could serve as a common coordinate framework, it would be an endothelial cell or a vascular muscle cell or an adventitial fibroblast, and so it's worth exploring. I'd say that the heart and vasculature, the diseases of these organs are of cell type and cell state. They're patterning defects. Congenital heart disease is certainly a developmental defect, and the efforts to look at embryos is going to be really informative there. Vascular diseases like atherosclerosis were like sort of the first cellular diseases, so we're sort of excited about that. Other organs also are impacted by their vascular effects, and there's kidney, lung, and gut and brain vascular diseases. Sort of a challenge and an opportunity is that vascular cells contact blood and so integrate the environment into their transcriptome. So how do we account for that? If we're going to get vascular cells from 50 donors, should we know a little bit more about their exposures or their lipid levels or those kinds of things, because that will be reflected in their single cell profiles. And then, like I said, vascular cells are abundant, and cell-cell interactions are really integral to this biology, like the endothelial cell immune cell interaction. So to kind of end, we sort of tried to postulate a little bit like what a first draft atlas would look like. And so we sort of settled on the first draft atlas for the heart would definitely be limited, we think, to post-mortem, brain stem death, unsuitable for organ donation hearts. And that, maybe that's just our opinion, but we thought that that was the best case of sort of a standard tissue source that would be as close to normal as possible. And then, we sort of, in the HCA white paper, I think it was postulated that they would use 20 normal hearts. And that could account for some inter-individual variability, but the truth is we have no idea. So I think it would be important to have an iterative experimental design, important to sort of freeze samples for pathology or for subsequent analysis and maybe reanalyze those samples at later times. But in the first 20 hearts, I think the main question would be, what is the inter-individual variability? And is our metadata capturing that? Are our sort of recordings of age, sex, gender and disease state, is that adequate? And then sort of like, I liked what the lung group said yesterday, is that the disease state actually could inform the normal instead of the other way around in some cases, because every heart's going to have some component of disease, and we don't know what that is beforehand. So in looking at sort of the extreme versions, like extreme cardiomyopathy or extreme atherosclerosis, we could use those signals to inform what is the extent of cardiomyopathy or atherosclerosis in the so-called normal heart. And then finally, extensive histology to validate markers and spatial location, I think, will be important. And then finally, the vasculature, I think a first draft atlas would be something like a large vessel, and then medium and small vessels would be sort of analyzed separately. And a large vessel would be surgical biopsy specimens from an aorta. So these wouldn't be postmortem samples that could be obtained. It would be relatively easy to get something like 50, 100 samples from the aorta. But medium and small size vessels will have different signatures, and those could be reanalysis of sort of tissue that we get from other single cell efforts, like the kidney, the lung. And all these cells and all these tissues will have some degree of atherosclerosis and variability from exposures, and so we'll have to account for that. And so the final thing I wanted to say is that spatial orientation was obviously a main topic at this meeting in general and in our small discussion group. And so we thought about different ways to do this. And so one way would be, you take the heart and you record distances from major structures, right? You just become very exact about this, 1 centimeter from the aortic valve or something. And that might be good. We pointed to some problems with that. Human hearts are just going to be very different sizes. So we could, one of the pathologists in our group suggested that we take a transverse section across the whole heart. Everyone just finds the midpoint of their heart sample and just goes all the way across and captures everything at that midpoint site. And that sort of was appealing because it could be something that could be standardized. We thought common coordinate frameworks would be hard to develop in human samples, but we could develop them in animal models, and the pig is evidently the most sort of reproducible animal relative to the human heart, even more so than, I guess, primates or rodents, certainly. And so that could be one possibility. And then we could also finally use preclinical imaging, like echocardiography, to assess for normal and diseased regions, and then sort of overlay that with the data that we get. So those are the points we wanted to make from our group, and we're happy to answer any questions.
So, great summary, and glad you had a good discussion. I agree that the heart and the vasculature are two different beasts that we evilly and maliciously put together on you. But I actually, I'm a big believer in the vasculature as a possible common coordinate frame, and I think that computationally, again, one would have to be smart about it, not look at the genome-wide distribution, but find the markers that can help with this. And, I do think there's a pretty mature idea that the immune cells need a separate protocol, and the immune atlas is going to collect from everyone and help everyone collect better immune cells from their own organ. But I actually think that the vasculature should present similar protocols, and we can get endothelial cells. And there's also, for example, the strong signature of just how big that organ is to where you are, how big the vessel is, as well as how far each individual cell is from the vessel, creating metabolic gradients that could explain a lot of the gene expression once we get into the spatial data. So I think both as where you are globally in terms of how wide your endothelial cell is, which is something I do think we can pick out of gene expression, and once we get spatial, how far away are you at little microcellular resolution from these vessels, determining gradients, is something extraordinarily powerful that the entire community should think of. So I actually want to punt my question to the other organs. How excited are you about using this as another piece of a common coordinate framework to interpret the data? And this is actually framed at the other organs.
Hi, I think this is a great idea, and we should definitely try and do it. The question is, how do we do it along the way as we acquire data?
So again, my idea would be that there would be some common group of both technologists to figure out how best to actually get these cells, because they're actually pretty tricky, finicky cells, to share protocols with the atlases, as well as computational people, who I know one sitting next to me is very interested in doing that, about developing computational methods to help do this. And we think it could be of great benefit if right here and right now people really think of this as another way to get some coordinate framework inside each and every atlas. But it would require sort of a centralized group, both on the technological and the computational side, to really facilitate each organ if the organs are excited to play along.
Yeah. Yeah.
Some more comments, questions? If not, we move on to the kidney. I think you forgot to say that heart is the most important organ. So next are Anna and Sam reporting on the kidney outbreak.
Okay. Hi, everyone. My name is Anna Greca. I am a kidney doctor at the Brigham and a scientist at the Brigham and here at the Broad. And I'm here with my partner in crime, Sam, to talk about kidney. So by way of a friendly competition, yesterday's folks were trying to sort of think about who has the more important organ. Well, we have the cooler logo, I think, and that's what I wanted to compete on, but this is actually the work of Catherine Blakesley in my group, so she's the artist, not me, and we just thought it would be cute to put it in the shape of a kidney. So this is what we talked about today. We had a great discussion. There was a lot of active participation, and we basically broke down the discussion and the project essentially in multiple pieces. And so Sam and I are going to alternate reporting out what was discussed. I just want to invite anybody who was in our session to jump in and keep us through if we are missing something major from the discussion. So just starting from the very beginning, we decided to just go around the room and catalog what activities may already be happening in the kidney atlasing space. And so there are actually some projects that are going on, and in some ways, there was a lot of foresight on the part of the NIDDK to start to fund some projects looking at the single-cell characterization of diseased kidneys. And so there's an effort called the Kidney Precision Medicine Project, which was represented by Matthias Kretzler here today. There's also an AMP program, also partially funded by the NIH, that also looks at some diseased kidneys. So KPMP is more focused on acute kidney injury and progressive kidney disease, which the most common form of that would be diabetic kidney disease, but just more broadly, chronic kidney disease. And then the AMP is focused on lupus nephritis primarily. But there are other efforts going on around the country. So there's some efforts going on at WashU, and Ben Humphreys was here today to tell us about that. UCSF, Minnie Sarwal was here. We have efforts going on at Jackson. Of course, we have ongoing efforts here at Broad as well. All of us trying to work toward a common goal of understanding the human kidney atlas.
So we spent a bit of time discussing what experimental strategies we may take, and discussed also what hasn't worked and what is more promising. There was clearly, everyone felt there was a need that we need some sort of standardization in terms of SOPs. We need to find a reference, an anatomical reference framework of how we take our samples and annotate that, and then also think about feeding that into the HCA database so that it becomes feasible to compare data sets. We had a discussion about frozen versus fresh, and there was a general feeling that sort of, I mean, they've obviously both got their biases, but they both work, and fresh is obviously, it's not necessarily the gold standard actually, but perhaps it's a starting point, and then frozen also will enable us to interrogate very large cohorts and also start thinking about disease. Yes, and then another thought that came up right at the end, and which we might get to, is to perhaps have a benchmarking exercise where a kidney is taken, biopsied let's say in the cortex several times, and then sent to different centers. People do their own sequencing, their own nuclear RNA sequencing, then annotate it, and then we can compare notes at the end to get a sense of how robustly and consistently we can generate and annotate data.
Yeah, and so then we kind of address the next part of this, which is, of course, the computational piece. So assuming that we resolve the experimental part, how do we integrate all of these data across different sites as well, and then also integration of metadata, which I know many of the groups have been struggling with as well. As with every other group, we had the discussion about cell types and cell states and the idea of sort of consensus markers in the community. And I think at the end, the only solution to that is to just continue to do this in an iterative fashion and draw from the experience of many different groups as we try to kind of arrive at our understanding of this. In many cases, a lot of our markers are coming from other species or from other modalities, but we can learn collectively and apply them as we try to truly map what a normal human kidney is comprised of. There was a point made, I think Sam made an excellent point about the fact that there's sort of an opportunity to work in a module in the kidney because the kidney, each of our kidneys is comprised of 1 million nephrons. So there's a unit of the nephron that is comprised of the filtering part and then the tubular part that results in the final production of the urine that is put into the bladder. So there is an opportunity to perhaps take advantage of this. So understanding the module may help us build the whole organ as we go along. There was another point that was made about electron microscopy is actually quite surprisingly standard for every clinical case where we do a biopsy of a kidney in a patient. And so we actually have great understanding and a wealth of information about electron micrographs. So at that resolution of being able to see cell-to-cell interactions in that spatial manner. And so perhaps we could be mining that data and even going back to those EM samples and getting additional sort of measurements to recreate the spatial relationships. There was also some discussion, and I know some other groups had that discussion as well, about the opportunity to take advantage of bulk data in order to annotate single-cell data and sort of integrate these data sets for the benefit of being able to better understand, again, what constitutes normal and getting greater granularity as to the different cell types. And so that was, I think, the bulk of our discussion on the computational side.
We then moved on about validation, and I think we all agree that to some degree that depends on what it is we want to validate, what technique we use, but one needs to use something orthogonal and we need to look at an independent cohort in one way or the other. And that's validating the RNA signal, but then also moving on from that and see whether that translates into function, and then use surrogate functions such as proteomics, metabolomics, and then also use that unique opportunity with the kidney that obviously the kidney is a site of physiology, and try to interrogate that physiology, for example, in these organ-on-a-rig system where one can manipulate a kidney, insult it in one way or the other, and then get a transcriptional readout of what that insult does. There's also the unique opportunity to sample the contralateral kidney in organ donors as a direct validation. And then finally, we should really utilize the clinical data that one gets with every patient. So not only age and sex, but also what the baseline kidney function is doing in blood pressure and so forth. So all the relevant parameters of kidney function.
So at the end, we sort of asked ourselves, well, what do we want out of a Kidney Atlas 1.0, the first version? And so I think that there was some consensus about the fact that we really need to address the variability between samples. And there were some ideas about how to do that. Some of it came in the experimental part. Some of it is in the study design. The idea of showing preference toward perfecting our protocols that we can all share for frozen tissue so that we can actually more easily create cohorts rather than being, we're able to run cells, but those come as they come whenever there's a procedure and tissue becomes available. So if we're going to create cohorts and more systematically study human kidneys, that may be an advantage, and specifically because we are interested in obtaining data across the lifespan, so from fetal or early childhood pediatric cases all the way to adults and then older adults as well, so the effect of aging. Of course the effect of sex and race as well. And that's where we also thought that there may be some opportunities taking advantage of some existing structures that are there where there's samples being collected in other parts of the world, including Africa and Asia, so that we may have a more representative sample from which to draw. There was also the idea of gaining better spatial understanding by performing, for example, micro-CT or some other such modality that will give us a better picture of the entire kidney as we embark on understanding a small piece of it at the single cell level. And then also the idea of integrating other existing imaging modalities such as histology or even electron micrographs, which, as I said, is sort of a unique feature of how we look at kidney tissue even in our sort of routine clinical care. And so these are just some of the first pieces that are coming together as to what we think would constitute the first attempt or the first version of this atlas. And I guess for next steps, we thought that we need to continue our dialogue, and so we hope to have a Slack channel for our community where we can actually bring in some additional voices. So one of the things that was on the earlier slides, we do think we need kidney pathologists to chime in because they have a much better understanding of the spatial organization of the kidney and the intricacies of the different parts of it. It's a very layered structure, very much like the brain with a cortex, a medulla, and many intricacies in those structures. And so perhaps input from the renal pathology community would be good. And I certainly know that I gain a lot from interacting with Astrid Waynes at the Brigham, who is the pathologist working with us on our projects. I think it will be valuable for the community. And I know that KPMP and other groups have also benefited greatly from having input from renal pathologists around the country, so I think that's important. So the Slack channel will allow us to engage with other members of the community, start to also look at sharing markers for different cell types, or starting to agree on these discussions between cell states versus cell types. And also, it's an opportunity to interact with the other communities that have ongoing work in this space. And I think it is wonderful if there could be some synergies in how we integrate metadata or clinical level data with our sample collections, a way to have a reference of where, which part of the kidney a tissue was obtained. These may be problems that are being solved in other communities as well. And so it will be important to be able to problem solve together and learn from each other. And so I think those were really our main points of discussion. And I just wanted to invite either members who were in our group or anyone else to really ask any questions at this time. Yes.
So I have two related questions. One of them, you mentioned cell types versus cell states twice. So my question to you is, what does cell type mean for you, and what do you want to use it for? And more generally, you gave an outline of what you want to collect for Atlas 1.0. But my question is, what would you want the Atlas 1.0 to answer for you? What would you hope to learn from it?
I mean, I think that anyone from our group could also chime in, but I think that the idea is to create the comprehensive catalog of all the different cell types in the kidney. The old, sorry? Defined types. I think that would be just all the cells that comprise a nephron, the functional unit of a nephron. In addition, it would be the immune cells, which we had quite a bit of discussion on. There may be resident immune cells that we could be interested in, even in a normal kidney, that may then inform disease states such as lupus, for example, which was discussed in our group. There's, of course, the vasculature. So there's many other cell types that are important. Endothelial cells are hugely important, and they're quite different in different parts of the kidney as well. So it will be important for us to understand that. Understand the cells that are important as comprising the nephron in the cortex versus the medulla of the kidney, and how they come together in the renal pelvis. So there's this spatial information that is important as well. But if you look at some of the old the old kidney physiologists microdissected nephrons and studied, the different parts of those. And so there's available information about different cell types from a long time ago, from many decades ago. And there have been, of course, many additional efforts to do this. And so we're looking at, upwards of, 26 more cell types in the kidney that we ought to be able to define. And in addition to that, we have all these other, pieces to it that we have to sort of assemble together. And so I think that we're getting there. We're starting to get there. My thinking about states, and this is something that the community needs to agree on, is states that may reflect different physiological perturbation, so to speak, which is, the kidney responds every day to all kinds of different things in the process of filtering and, adjusting things like blood pressure, adjusting things like, hormonal regulation of various things, secretion of erythropoietin is done through the kidney, bone mineral metabolism is controlled by the kidney. There are many states that one could imagine one could capture, and that's where multiplexing data from proteomics and metabolomics, especially metabolomics, could potentially be helpful. I think that we're just scratching the surface of that, but that could be informative of states in addition. And of course, that's not even going into disease, where now you have major interactions with the immune system that we need to understand, and again, some ongoing efforts in lupus and other places. And I think that that's just going to build from there. But being able to understand the fundamental composition of the nephron and its interactions with other cell types around it that are still part of this integral organ, the kidney, like the vasculature and the immune system that's there, for starters, I think will be important through the lifespan and across gender and sex. So I think, we already have some ideas that, for example, men and women have a slightly different composition in the proximal tubules of the kidney. It seems that that's, from clinical information, that seems to be true from just basic histology. So wouldn't it be fascinating to be able to really characterize that at the level of the single cell for our atlas? I think that would be fantastic as a goal, as a first 1.0 goal, and with much more work to be done afterwards. Any other questions or thoughts? Anything I missed from any of the people who were there? Anything that?
If not,
thanks,
Anna. Thanks, Tim. Also for opening the logo competition. So on the next HDA meeting, we have a logo breakout session for logo design. The next is Gary on the reporting on the liver.
Okay, great. I'm going to tell you why the liver is interesting. I don't know if it's we could say it's the best, but the liver is not just a big bag of hepatocytes. That was one of the comments that we got in the last one of the last HCA meetings. Some interesting things. Interestingly, liver is formed of repeating units. This sort of hexagonal lobule here is repeated about 1 million times in the liver. It's about 1 millimeter or so in diameter. And then there are small capillaries in here that do the work of filtering the blood that are lined with different cell types and have a fairly well-defined structure. The liver is a metabolic factory, over 500 functions known. It's an immune center. Most of the macrophages in the body are in the liver. It's got an amazing regenerative capacity. You could remove most of it and it will grow back. And one of the interesting things that I learned interacting with liver community is that there are major liver diseases that are increasing in prevalence. And for instance, non-alcoholic fatty liver disease, or NAFLD, affects 20% of the world's population. It's like 1.7 billion people, and it's just getting worse with diets and issues with how people eat, etc. Viral hepatitis affects hundreds of millions of people, and so this is pretty. And there are no treatments to end-stage liver failure other than transplant. So. And the rate of these diseases as they're getting bigger is going to lead to more transplant needs, but you actually can't solve the problem that way. So we need to understand the liver to develop therapies. Okay, so there are a couple of maps have been published. The Itzkevitz lab published 2 groundbreaking maps in mouse over the past year, and Sonia McParlin, Ian McGilvery, and myself published a human liver map just last week with 5 human liver samples. And so these are out, so that's great. So we have our first draft map. The liver community is excitedly coming together. There are 9 liver projects that we knew about before that we're already talking to. all of these people are talking to each other. It was great that everybody, at least one person from every one of these projects was able to attend this meeting today and is discussing about how to move the field forward. And we identified additional projects as well. So there's about 10 different projects that are active for liver mapping, representing a range of disease types and cell type interests. There's a fetal map, there's a nascent liver map project that hasn't collected samples yet, but as we identified samples, and the rest of them are focused on adult. Some of the interesting discussion points that were raised at our meeting this morning is that different people are working with different types of tissue collected in different ways. So we've worked with the transplant team to get a whole caudate lobe, and there's a protocol that is useful for that to get to that, was used for our map. But other people are working with biopsies and normal adjacent tissue from tumor resections, and everybody's developed their own protocols. So there's a big need among our community to compare these protocols. But we also realize that different people have different cell types that they're interested in, and these protocols are going to be different for different cell types. So we realize that there's not going to be one protocol that we're all going to use. There are going to be multiple protocols, and they'll be tuned for studying different cell types. So for instance, immune cell types are very robust, it seems, and hepatocytes die at the slightest bit of processing. So there needs to be, most protocols will work maybe fine for immune cells in the liver. There are some specific challenges that people are facing with experimental processing of the tissue. As was mentioned, for cardiomyocytes, liver hepatocytes sometimes have 2 or more nuclei. There's a huge mitochondrial level, like 2,000 mitochondria per hepatocyte, and these need to be, and there's also fibrotic liver samples that need to be dealt with separately. So one of the interesting ideas that came out of the discussion was that a lot of people have been talking about cell types and cell states from a biological function point of view, but we also need to think about this cell types from a practical experimental point of view. People were interested in knowing how they can identify whether they've got, how well their sample prep is working. So it would be nice to have a database, for instance, of experimental signatures that people see are associated with certain cell types, like certain transcript types and library sizes and other viability rates and other things like this that could go into a database of experimental factors that people identify. So when you do your analysis, you can know whether you are your rates of viability, for instance, are in line with what other people see on a per cell type basis. So so Dana Pear mentioned that there's that might be an idea for a portal to look at a range of signatures that are various different views of signatures of cell types. We also, it's absolutely important because of all of these problems with different cell types to look at single nuclei sequencing, and so one of the big things that we'd like to do as soon as possible is compare single cell and single nuclei maps. We realized that many of the samples that we get are inflamed, so there's a discussion of healthy, normal, and what we actually get from tissue samples. Neil Henderson was mentioning that there's ischemia effects, and so we need to we very likely need to look at model organisms to help define what's normal and what's healthy, and be able to control the rate of inflammation or response to drug toxicity that might be actually widely prevalent in the human population. So I like what was mentioned with a couple of points earlier about how we need to study the disease situation to understand the healthy. In this case, it makes a lot of sense as well, because the healthy population is actually maybe frequently inflamed. So maybe half of the samples that we have are actually inflamed at a normal amount. So we have to study the extreme inflamed conditions to understand the inflamed factor, and also model organisms. Spatial is important. Liver for decades has understood that gradients are vital to the function of the liver. So as the blood flows through the capillary, there's a gradient of functions, and the cells actually act differently and have different gene expression profiles. And so we need to incorporate spatial technology. One of the questions is, we have 1 million repeating units. Can we just study one, or do we need to figure out if they're different in different places in the liver? The draft answer that we felt is that no, we can just study one for now, but it's not well known, so we do need to check. But there is preliminary results suggesting that the lobules are pretty similar across the liver, which is very nice, because that means that we can study this one section and extrapolate it across to the whole tissue. So it would be really great to have a 3D model of the liver where we know we've mapped all the vasculature, which I like the point about the vasculature coordinate system, and then attach these lobules to the vasculature and make a full 3D map. So that would be great. Dana mentioned that we could share technology with the Space TX project, so we'll be looking into that. We need to link with the immune map because there's a lot of immune cells in the liver. And one of the interesting things that was raised is, we've talked a lot about the immune map. Immune cells are present all over in many different tissues, but endothelial cells, which are important in the liver, are also present in lots of tissues. So maybe we should have an endothelial map, even though that's not about liver. Endothelial cells are very important in the liver, and it's just an interesting idea that came out of our discussion group that could be food for thought for this community. Finally, just another practical little point was that a lot of people are developing site-seq antibody panels, and there are some standard ones that you can buy commercially. But we felt it would be nice if we could harmonize, share what panels people are developing and using, and even there was an idea that you could harmonize the antibody barcode. So if one person uses an antibody, if multiple people are using the same antibody, they all use the same barcodes. And so there was some discussion that could maybe happen through companies, but another thing to discuss in our community. So finally, we got agreement to share 10 new protocols on protocols.io. So there are 2 liver protocols that exist right now, but around 10 people said that they're going to post their protocols in the near future, so that's great. We also decided that we're going to work together on a combined analysis and map once multiple data are published. And also there's a lot of interest in forming a protocol comparison working group. So it would be a great idea to have a protocol comparison paper that comes out of that. And then we didn't discuss it in our discussion. I forgot to mention it, but other discussion around this meeting has suggested that maybe we should organize a single-cell liver conference, and there was some discussion in the organizing committee about the importance of these. So that would be, and everybody would be excited about that. So we're already thinking about how to do that. So Maz Hanifa, Neil Henderson, and Sonia McParland were the other leaders of our session, and Martin Williams and Charlotte Scott were the scribes. So if I missed anything, please mention that in the question session. Thanks.
So a lot of the groups throughout the past 2 days have been talking about making sure you get your right type of cells, looking at viability, looking at cell viability and cell fraction. What I haven't heard is making sure that your cells are unaltered and that the protocol doesn't alter your cells, particularly in the case of immune cells. You can keep them alive and happy looking or alive looking, but particular handling actually changes their gene expression. So I think viability and live dead should not be the only criteria by which we measure a protocol is good for a cell type.
Absolutely. Yeah, so we need to catalog these ideas and, define what we need to measure.
About the lobules and the repetitiveness, because that was a question that also arose in the gut session and in the kidney session, right, on the repetitiveness of the units. So you mentioned that there are some preliminary notion around this? I think it would be very helpful across the board to get
Yeah.
some answer around questions, and it might not port from one tissue.
So there's some discussion that work in mouse has shown when you take these lobules from different lobes of the liver, the gradients and their gene expression patterns are the same, basically, so from the Itzkovitz group. So that's the data that I guess we have so far. So that would suggest that we could, but there is some information that comes from liver disease that suggests it might not be exactly like that. So people know that the liver disease, certain parts of the liver are affected more preferentially in certain liver diseases. So that might suggest that it's not exactly uniform, but as a first draft, I think we could say reasonably well we would just study one and
But would you benefit if they are actually uniform or not? Would you benefit more from doing one, or if they're all the same, you could sample less from any of them, and you would still get sort of the same answer, but you might also then identify some variation that right now you're not, possibly is not there, but possibly is underappreciated.
So I think,
I'm just trying to figure it out.
So personally, I think it would be great to study the lobule very carefully, but
maybe spread some joy around.
So we could, but then also, as you just said, we would want to we want to check that things that we think are constant, look at the variation of those things. So you could do that in a few different ways. You could study carefully lobules from different parts of the liver, and you could also study a lobule, and we'll have information from multiple different samples no matter what. But specific features that you identify, like a gradient, gene expression across a gradient, does that feature hold in different places?
Yeah,
or the cell type composition, does that feature hold in
right,
different places?
because the gradients could hold in, right, within a lobule you could have the same gradients per Shalev's work. That actually does not rule out the possibility that they're still different from each other.
Yeah.
Right?
Yeah.
So I think that's the piece that we have to be careful about.
But we felt that, yes, it's important to figure that out, but it doesn't hold us back from going deep on one and trying as a first draft, say, this is what we'll extrapolate from one, but then let's check and we'll update. So I think that makes sense for us.
Yeah, I mean, you can imagine factors where some of these gradients, which involve particular genes and processes, are very repetitive for this factor, but then there are factors that determine differences across the organ, and you could imagine both of them coinciding.
Absolutely. Any other questions?
No more questions.
Okay, thank you.
Big applause to all the speakers. So I think we had a great meeting, and now it's time to wrap up. I will hand over to Aviv and Sarah.
Actually, 4 of us. I'll turn this off. It's actually 4 of us, also Piero and, yeah. Okay, so how do we advance this slide? So I'll start by saying I think it was a very exciting meeting, and in my view, the breakouts were of a very different nature than previous breakouts that we've had. You can see that this is an ongoing conversation rather than the first time that you get to look at a problem and meet people, and that this is coalesced. And as we said, I think also in the beginning, the basic tools are now there. They've been tested and tried. People have data. They know it works for them. And now the question is, all lobules or just one, and there's even consensus that's emerging in each of the organs. We had hundreds of remote participants. It was actually a lot of fun, I think, to monitor that when you were like on YouTube and on Twitter and so on. There was a lot of active engagement from them, and that allowed us to have a meeting that's kind of small and open at the same time, which is a new model that we've tried in this meeting and that we plan to do in the past. We've seen very detailed updates and very specific plans for the lung and the gut and the immune system, the heart, the kidney, and the liver. These are not all the organs and systems that people are studying now in HCA, so there's a little bit of a rotation between them now across the meetings. And one of the ideas that came up actually initially in a meeting together with the funders, but I think is really taking hold today, is that we could start having meetings for specific organs, not as the general meeting, but separately from it or attached to it, so that we can dive in and make that each community really coalesce and report to each other as we progress in this journey. Some favorite tweets. I thought the one from Naftali Kaminsky was very lovely because he juxtaposed how nice it is here to how difficult things are in the general world. And so we hope that some of the HCA spirit will expand. People were excited to see everything. The ethics, for example, was highlighted by a participant. The protocols I/O, the sharing, and so on. So it was a cool meeting. And the one kind of in the middle, this one, is a grad student. Apparently yesterday was a national holiday in Belgium, or a public holiday in Belgium. And here below is our live stream. Here above is our live stream, and here below is the grant that the student was writing. And so they found it to be a very productive day. With this, I'll turn to Sarah to maybe say a few things. I think this is how you advance the slides.
So I just want to say a huge thank you to the Broad team, to Aviv, of course, but also to Jane Lee, and who's been amazing from the HCA Executive Office here, to Tracy Andrew from the Executive Office in Sanger, to Masaki Furuno and Miho Ito from Japan, from Riken, from the HCA team. And also massive, massive big thank you. I know it's been a tremendous effort from Christine Schwenk and Elliot Boblet working with her. And I think we should just give that team of people a massive thank you. So there have also been people involved as scribes and also moderating the chats that have come in, and their names are Ella Mazizi, Katie Geiger, Jody Hirschon, Jane again, Kirsten Meyer, Jenny Rood, Ashwarya Subramanian, Pratiksha Thakori, I'm not sure if I've got all the names right, and Rosa Vento. And let's say a big thank you to all of them as well. And without further ado, I will hand over to Piero.
Okay, thank you, and I'll be short. But this meeting was great, but it's not the final meeting. So we are planning the next stage, and actually, so please keep save the date. So we're gonna bring you to Japan. So this is a picture from Jay. Actually, has been analyzing how people move in Shibuya, because even if you don't know what is the most complex organ, we know that Tokyo is the most complex city. So we are the Disney of how people move. That about 30 million people in Tokyo now, plus many subways. So it will be a very interesting organ to study. Anyway, keep in mind those dates. We have a workshop. Before we just heard that EMBO is happy to support us, and then the Human Cell Atlas. Tokyo is not only science. We have a lot of food, traditions, many places to see, nature, and also we have a Chung that is, he was here, that he promised to take you to a bath with the monkeys. Actually, it is a true picture that, I mean, in some places you can see that. And also like to thank all the speakers that have accepted already to talk there. Many of you are in the room. Thanks a lot, and see you in Tokyo as many as possible. Thank you very much.
Thank you, everyone.
Thank you.
Thank you.
How many hours?